Hi students, am I audible to you all? Hi everybody, am I audible? Hi students, am I audible? So hi everyone, good evening, welcome to Unacademy NEET English. I'm your biology educator Ambika Sharma, and today we are going to start the first chapter of NEET Endgame, and that is locomotion and movement. So how are you all? Do let me know. Am I perfectly audible and visible to you all? Yes. Am I perfectly audible and visible to you all? Yes, I believe I am. So good evening guys, welcome. Hi Stella, Someshwar, Isaac, okay, call me Bablu, Thirumalai, so Snover, hi. Hi, how are you guys? How are you? So guys, we have started the NEET Endgame, and you know that here we are going to revise all the chapters, and these one shots will not exceed 5 hours, right? They will be less than 5 hours. They will be somewhere in between 3 to 4 hours. We have to complete one chapter. But because we are completing that chapter in 3 to 4 hours, are we going to compromise with the content? The answer is no. But here we will focus on the most important things. Okay, so if you are doing any chapter for the first time, yes, you can rely on the series, right? But the point is, if you want, this is my suggestion, that if you want to understand a topic from scratch, that is from zero, you can also go for our Game of NEET series, the most popular series, right? You can also go for what? You can also go for Game of NEET series for the very detailed, for the very detailed revision of any particular chapter. But let me tell you, right, that you have to do for the most important chapters only, right? That you have to do for the most important chapters only. Okay. So now let's start, guys. Now let's start. Ma'am, please read the, please, I need to qualify NEET. I need 200 plus marks and not studying here. I'll be able to score 200 by focusing on biology only. Yes, Neha, definitely. If you are targeting 300 marks, yes, you can just score the marks from the biology only. But still, I will suggest you to go for the most important chapters of physics and chemistry, at least most important topics of physics and chemistry, right? Just to ensure your marks. So guys, I'm going to start the session. So quickly hit the like button if you're new to our channel, subscribe to our channel, guys, and let's start. So excited? Yes, excited for the class? Excited for the class? So guys, just one free advice for all of you: do not underestimate the biology. We are here in the month of February, right? In the month of May. On 4th of May, you guys are going to have your exam. So please do not underestimate biology. Do not underestimate biology. Okay. So if you are thinking that, nahi ma'am, I will just study in the month of April and I'll be able to score 300 plus marks, I'm sorry, it's a big no. You have to study the biology daily. You have to practice the questions daily, and you have to revise. You have to analyze the mock test, and you have to work on your errors. Okay. Okay, so let's start, guys. Let's start the chapter, right? Locomotion and movement. And if you want to be the part of our dream crash course batch, in which again we are discussing the human physiology, then here you people can see, you will get the link for the dream crash course batch in the description box of all the videos that are there on our channel. Okay, so you will get this course just at ₹2,999, right? The weekly test, proper revision, everything will be there. The DPPs will be provided to you, and we will complete the syllabus in 70 days. Okay. Okay. So without wasting any time, be the part of the batch. Now let's start the class. Let's start the chapter locomotion and movement, and here we will go as per NCERT. Okay. Okay. So let's start. So what do you want to start first? Human skeletal system or muscles? Right? We divide this chapter into two parts now. We have the muscles portion and human skeletal system. So what do you want to start first? Please tell me quickly. What do you want to start first? Yes, please. Yes, please. Yes, please. Yes. What do you want me to start first? You want to start the muscles part first or the human skeletal system first? Muscles, skeletal system, muscles, skeletal, like Although you are interested in both skeletal and muscle, so obviously we have to cover the entire chapter. So let me tell you, students, when you talk about the muscles part, right, what is the most important thing? See, first of all, you should know the difference in smooth muscle, the cardiac muscle, and the skeletal muscle. That is important. The another important thing is the sliding filament theory. It is one of the most important topics. So if you will solve, right, the PYQs of this chapter, you will get maximum questions from the sliding filament theory. Okay, and when it comes to human skeletal system, trust me, it's very easy. It's very easy to remember the bones, right? Just one time you need to be focused, and then with, you know, quick revisions, you can master this topic. Okay, you guys can master this topic. So let's start. So let's start from the starting itself. Let's start from the muscles part itself. So the word here is locomotion and movement. Movement, right? To move from one place to another. Right, overall I'm saying movement, to move from one place to another, to move the forelimbs, hind limbs, move the eyebrows. These all things are what? They are the movements. What are these things? They are the movements. But when we are saying locomotion, locomotion, location, locomotion, location. So when, because of any movement, when there is a change in location, or you can say that the movement due to which there is a change in location, we call it as locomotion. What do we call it? We call it as locomotion. Is that clear? What do we call it, students? We call it as locomotion. So what is the meaning of locomotion? Locomotion means that the movement due to which we are able to change our location. Is that clear? Is that clear? The movement due to which we can change our location. Understood? Yes, please. Understood? So this is about the movement here. Okay, this is about the movement. So locomotion is what? It's a type of movement. Locomotion is what? It is a type of movement. Is that clear? What is it? It is a type of movement. Is that clear? Is that clear? So when we talk about the types of movement, so I'm writing it here. Movement. Yes, I'm writing it here. The word here is movement. So we have muscular movement and non-muscular movement. What do we have here, students? We have muscular movements. So when we are saying muscular movements, of course, we are talking about the movements in which muscles are involved. And there are movements in our body where muscles are not involved. That will come under non-muscular movements. That will come under what? That will come under non-muscular movement. So make sure you respond here in the chat. So muscular movement and non-muscular movement, these are the two words. These are the two words. So when it is the muscular movement, obviously we are using the contractile property of muscles, right? Contractile property of muscles, which is used for locomotion and other movements. So what will be used here? Contractile property of muscles, right, which is used for change in location and for other, yes please, and for other movements, right? Other movements. Can you quote any example when we are saying other movements? Right? When we are saying other movements? Yes, please. So contractile property of muscle, which is used for change in location and for other movements of human beings and some other, right? And some other multicellular organisms, right, bache? Some other multicellular organisms. Like here you can consider the movement of jaw, movement of tongue, right? Normally we are moving our forelimbs, right, bache? So movement of limbs, movement of jaw, movement of tongue, all will come under, yes please, all will come under the muscular movement. All will come under the muscular movement. So now when you are talking about the non-muscular movements, what do you have? You talk about the ciliary movement, right? The cilia that helps in the movement, of course. Flagellar movement is there, right? Ciliary movement is there. Flagellar movement is there. Amoeboid movement is there. Now you have to quote the examples here. Yes, please. You have to quote the examples here. And then you also talk about the cyclosis, that is cytoplasmic streaming. So when we are talking about the non-muscular movements, this is what we have, right, people? This is what we have. So we talk about the sorry. So we talk about the ciliary movement here, flagellar movement here, amoeboid movement here, cyclosis here. So what is this cyclosis? Bache, cyclosis is the cytoplasmic streaming, the movement of cytoplasm. So where do you see this? Where do you see this? See, when you talk about a eukaryotic cell, the cytoplasmic streaming is there. Cytoplasmic streaming is there, which is again very important, which is very important for the intracellular transport, which is very important for transporting the proteins or something else from one part of the cell to another, right? So that will come under the cyclosis. That will come under the cytoplasmic streaming. And where do you see this? Where do you see this, students? You will see it in the eukaryotic cell. Where do you see that cyclosis or cytoplasmic streaming? It will be shown by most of the cells. You can say that particularly your eukaryotic cells. So it will help in the distribution of material. Is that clear? Is that clear? Yes. Is that clear? Tell me. Tell me. So now let's talk about the ciliary movement. So can you give me any example here? Can you give me any example here of the ciliary movement? Cilia, some small hair-like structures. So I'll give you the example of paramecium, right? I will give you the example of paramecium here. 1 second. You know na paramecium, slippers organism it is, right? Yes, you know paramecium. It's a unicellular eukaryote. It belongs to kingdom Protista if you remember. Yes, it belongs to kingdom Protista if you remember. We are talking about the ciliary movement here. Right, I'm giving you the examples of ciliary movement here. Yes, Piche. So when you talk about the ciliary movement, this is the cilia that we are talking about. So you know that it helps in locomotion as well. It helps in changing the position as well. Plus here, it will also help the food to enter here into the mouth as well. Ciliary movement. Now when you talk about us, right, if you remember our trachea, in the case of our trachea, we have that pseudo-stratified, yes, do we have that pseudo-stratified ciliated columnar epithelium, columnar even glandular epithelium? Do we have that? Yes or no? Do we have that? Yes or no? Yes, ma'am, we do have. We do know about it. We study about it. Pseudo-stratified. So what is the meaning of the pseudo-stratified? Pseudo means false. Stratified means layered. Means it's a single layered structure, but it looks like multi-layered. Plus it is ciliated. The cells are having cilia. The cells are having cilia. Columnar means cells are tall and cylinder. Nucleus is present at their base. Glandular means secretory. That type of epithelium. So cilia is lining it. Cilia is lining it. So cilia will help in the movement. Just for an example, if there is any dust particle trapped, so mucus, it will be there in the mucus, and cilia will help in the movement of that mucus loaded with that dust particle and will throw it out. Right, we'll throw it out. These are the examples. These are the examples. You can take the example of fallopian tube as well, right? Fallopian tube as well, right? So in the case of fallopian tube, you will see the ciliated epithelium is there, and that ciliated epithelium will also help in the movement of egg. Yes, please. Do you remember this? Do you remember this? So cilia and trachea, right, will help in the reproductive tract. This is what you are going to see. And I'll give you the example of flame cells. Do you remember flame cells? Protonephridia, also known as solenocytes. It is protonephridia. It is also known as solenocytes. What's that? It is also known as solenocytes. So flame cells, they also have cilia. So flame cells, where do they play the role? They play the role in excretion. So that's how the ciliary movements are. Now, let's talk about the flagella. Let's talk about the flagellar movements. So in the case of sperm, we discuss about the flagella. We discuss about the flagella, right? So, of course, that's what we will discuss, right? Sperm is also having the flagella that is there for the movement, isn't it? Isn't it? Isn't it? And that's not the only thing. That's literally not the only thing. There are other examples also. There are other examples also. Can you give me any example? Yes. Can you give me any other example? Any other example? Flagellar movement I'm talking about. Anybody in the class, see, it's a one shot now, right? It's a one shot, and it's a very quick wala one shot. So I believe that students, they know the answer of certain questions, right? So quickly tell me this cell. What do you know about this cell? What do you know about this cell? Anybody here in the class? Very good. Jashan Bache, very good. So obviously we are talking about the collar cells here. Collar cells, flagellated they are, right? The choanocytes basically. Choanocyte in the sponges, in the porifera. We discuss about the choanocytes there. We discuss about the choanocytes there. They line the spongiocele, if you remember. This is a direct line from the NCERT. So that flagella movement will maintain the water current, if you people remember, right? So this is also the example of the flagellar movement. Right, it is also the example of the flagellar movement. You take the example of your spindle organism, right? Spindle organism. Spindle organism is what? It is euglena, having two flagella. One is short, one is long, right? Having two flagella. One is short, one is long, right, bache? So this is the spindle organisms, right? This is what? This is the spindle organism. And here also we will see the flagellar movement. So these are the example. So when you talk about the amoeboid movement, my dear students, yes, when you talk about the amoeboid movement, my dear students, remember your leukocytes, right? Your WBCs, your macrophages, right? Your WBCs, your macrophages, they show the amoeboid movement. The irregular shape is there in the cell. So amoeboid movement means the pseudopodia formation will be there, right? The pseudopodia formation will be there. Okay, bache. So this is about the amoeboid movement. This is about the amoeboid movement. Now here in this chapter also, we'll get to know about it. But still, I'll tell you one thing here, that when you talk about the amoeboid movement, you talk about what? You talk about the pseudopodia. That's what we discussed. Pseudo means false, podia feet, false feet, it is pseudopodia. So when you talk about that pseudopodia formation now, students, right? So you will see that pseudopodia will form, right? Basically here you will see, what will you see? You will see the cytoskeletal element. Which cytoskeletal element it will be? Actin filaments, also known as microfilaments. They help in the pseudopodia formation. Right? Just in the case of eukaryotes, cilia, flagella, they have microtubules. That is the cytoskeletal element. 9+2 arrangement. That is also the cytoskeletal element. And here, this is what we have. So you can see the thing here, the direction of movement, the substrate, right? The actin network, actin filament. As I said, they are also known as microfilaments. They are also known as microfilaments. So here you people can see, right? So this actin will help in the formation of the pseudopodia. Understood? Understood? Chaliye. So let's quickly see the things from the NCERT. Yes, bache, we have covered the NCERT here. Let's have a quick revision of that. So they are saying that streaming of protoplasm, I told you already. Streaming of protoplasm, it is cyclosis. It is cyclosis or cytoplasmic streaming. Or what is it? It is the cytoplasmic streaming. That's the word that we use. Okay, that's the word that we use here. Okay, so it in the unicellular organisms like amoeba, it's a simple form of movement. Okay. Then cilia, flagella and tentacles. They are also shown by many organisms. So when you talk about the tentacles, remember Hydra, which is having tissue level of organization. So tentacles are there. Tentacles having nematocyst. Remember this? Hydra, which is a polyp. Then human beings can move limbs, jaws, eyelid, tongues, told you already. So some movements that help in change or that result in change of location or place, that is known as locomotion. So locomotion is a type of movement. Locomotion is a type of movement. Is that clear? Locomotion is a type of movement. So walking, running, climbing, flying, swimming, they are the locomotory movements because the location will change due to which. Okay, bache? Okay, right. Now paramecium, I told you paramecium, cilia will help in the movement of food through cytopharynx and it is also helping in the location as well. Hydra uses the tentacles for capturing its prey, right? And tentacles are also used for locomotion. Okay, so here remember they are having some stinging cells, your nematoblast cells. Nematoblast cells. Is that clear? So we use limbs for the change in body posture and locomotion as well, right, bache? So all locomotions are movement, but all movements are not locomotion. Just for an example, I'm moving my limbs like this, right? I'm moving my limb like this. Okay, I am doing what? I am moving my limb like this. So what is it? It's a kind of movement, but it is not changing my location. So it is not locomotion. Okay, it is not locomotion. Understood? Understood? So I hope this part is clear here. Bagi, why this locomotion is required? First of all, locomotion, methods of locomotion performed by animals, they vary with their habitat and the demand of situation. Like birds, they fly, we know it. So locomotion is for the search of food, shelter, mating, right, suitable breeding grounds, favorable climatic condition, and to escape the enemy. So now let's talk about the types of movement, and of course we have covered it already, right? We have covered it already. Is that clear? Is that clear? So examples here are important. Macrophages, the phagocytotic cell, leukocytes, your WBC, right? They show the amoeboid movement. They will form pseudopodia. How? By the streaming of protoplasm. And these elements like microfilaments are also involved in amoeboid movement. So this question is important. It's a PYQ, right? It's a PYQ. Okay. Then is the ciliary movement, right? It occurs in most of our internal tube-like organs, right, where the ciliated epithelium is present, right? So here the cilia movement, I told you, I have given you the example of trachea bache, I've given you the example of female reproductive tract, right? And the movement of limb, jaw and tongue, it is the example of muscular movement. So when it comes to the locomotion, it requires a perfect coordinated activity of muscular, skeletal and neural system. So when we are saying locomotion, what are we going to see? What are we going to see? In the case of locomotion, there is a perfect coordination. There is a perfect coordinated activity of muscular, skeletal and neural system. These three systems, they will be involved. Any doubt? Any doubt here? Yes? Any doubt here? Okay, so let's discuss about the muscles. Let's discuss about the muscles. So we have studied that cilia and flagella are the outgrowths of cell membrane, right? They are talking about the chapter cell. So again, I'm repeating this part. When you are talking about the eukaryotes, right, that flagella is having microtubules. That microtubules are made up of what? That is made up of tubulin protein. Which arrangement are you going to see here? 9+2 arrangement. Remember? 9+2 arrangement. But on the other hand, when you talk about the flagella, in the case of prokaryotes, like in the case of bacteria, so it's a very simple structure. It is made up of flagellin protein. So this is for your quick revision. That's how you have to relate one concept with another. That's how you have to revise the things. Okay, that's how you have to revise the things. Okay, okay. Done? So flagellar movement helps in swimming of sperm, maintenance of water current in the canal system of sponges, locomotion of protozoans like euglena, right? So now let's talk about the muscle. So what are they saying? That muscle is a specialized tissue of mesodermal origin. Make sure you write the points here, okay? Do write the points here, okay? Right? Try to make the short notes with the class itself. So we are talking about the muscle. What are we saying, bache? That they are mesodermal in origin. What do we say? That they are mesodermal in origin. Now, what's the meaning of being mesodermal in origin? Means it is made up of which germ layer? It is made up of mesoderm. It is made up of mesoderm. Mesoderm is what? Mesoderm is a germ layer. Now, do all the muscles, they are made of mesoderm only? No, exceptions are there. So when you talk about the exception, you can discuss the muscles of iris. So iris muscles, they are ectodermal in origin, right? Iris muscles, they are ectodermal in origin. Okay, they are what? They are ectodermal in origin. So yes, this is an exception and you should know about the exceptions, number one. So what are we saying? We are saying that when we talk about the muscle, it's a specialized tissue of the mesodermal origin, and 40 to 50% of the body weight of human adult. 40 to 50%. Yes, 40 to 50% of the body weight is contributed by the muscles, right? 40 to 50% of the body weight, it is contributed by muscles. It is done by what? It is contributed by muscles. That's what you have to remember. Now, we have to discuss the properties of the muscles. right? What we need to discuss? We need to talk about the properties of muscle. So please write down the properties of muscle in the, yes, quickly in the chat section. Do let me know the properties of muscle. Yes, please do let me know the properties of muscles. Quick. It's a very simple question, right? It's a very simple question, and I believe you know the answer. Yes. Right? Yes. What's that? It is CEEE. It is CEEE. So first is contractility. So what is this? The ability of muscles to contract or to shorten. What's that? It is the ability of muscles to contract. So when they contract, they will shorten. So that's why we write it like this, to contract or shorten. So this is first, right? That is contractility. Then excitability. So when we are saying, bache, excitability means muscles, they respond to the stimuli, right, bache? The muscles, they respond to what? They respond to the stimuli. Okay, the ability of muscles to respond to stimuli. Is that clear? That is the excitability. Then we have extensibility and elasticity. So extensibility means the ability of muscles to stretch. Yes, the ability of muscles to stretch, right? And here elasticity means to come to the original position. That ability of muscles that they can even come back to their original position. Is that clear? That they can come back to their original position. So this is the property of the muscle, contractility, and 3E means excitability, extensibility, or elasticity. So ability to stretch is extensibility, and ability of muscle to return to its original length after stretching, right, is basically what? It is the excitability. It is the elasticity. Understood? So see this, these 4 words are written here. These are the special properties of the muscle. You have to remember it, right? So on the basis of location, appearance, and nature, we have 3 types of muscles: skeletal, visceral, and cardiac. Out of it, the skeletal muscles, they are voluntary. We can control them with our own will, but visceral and cardiac muscles, they are involuntary. What are they? They are involuntary. Is that clear? So let's discuss that. So skeletal muscle fiber, obviously they are associated with our skeletal system. So they occur in limbs, body wall, face, neck, etc. Cylindrical in shape, multinucleated muscle fibers they are. Right? They are not uninucleated, they are multinucleated. Right, bache? So I'll mark the main point. Fibers are unbranched here. Right? I'll mark the main details here. Fibers are unbranched here. They are multinucleated. Matlab since CTM is there, right? They will soon get fatigued. You know na, let's say you do not run daily, right? Today if you will start running, right? Next day we have the pain, now we have the cramps. What is that? What that cramps are? They are basically the accumulation of lactic acid. That is the fatigue, right? What's that? That is basically the fatigue. That is basically the accumulation of lactic acid. Lactic acid is accumulated, right? Because in the muscles, what was there? Fermentation was there. Lactic acid formed. Okay, lactic acid formed. So lactic acid will result in the fatigue. Okay, that is the fatigue. So the skeletal muscle. So when you talk about the skeletal muscles, right, they are associated with our skeletal system. They are very important for this movement of the skeletal system, and they soon get fatigued. They are multinucleated, they are unbranched, and they are voluntary in action, right? They don't have intercalated discs. I'll tell you what intercalated discs are, right? And this skeletal muscle fibers are also known as striated muscle fibers. They have the striations, right? They have the dark or light bands. We will discuss about its structure. Okay. Okay, now smooth muscle fiber, as the word is smooth, they have no striations, right? No striations are there. No striations means no that dark band, light band is there. They are spindle shaped, means fusiform. Okay, so they line the hollow organs of our body, just say your alimentary canal, right? In the alimentary canal, in the walls, you will see this. In blood capillaries, you will see this. In the uterus, the middle layer, myometrium, is having the smooth muscles. So what is the function of smooth muscles? First of all, they are smooth, they don't have striation, they are fusiform, they are uninucleated, right bache? They are uninucleated. The nucleus is in center, right? They are uninucleated. Their fibers are also unbranched, right? So myofibrils are present there. We'll discuss the ultrastructure of muscles. We'll tell you what myofibrils are. So myofibrils are without light and dark bands. Myofibrils are without light and dark bands. Is that clear? Is that clear? So these are the muscles which do not get fatigued. Like in their case, the fatigue is very, like it's not very quick, just like skeletal system. So they do not get fatigued, right? Or you can say that is something very slow process there. Okay, and again, they are also involuntary in action, smooth muscle fibers. So location should be clear. So hollow organs of our body, they are lined with, right? That's a question that was in the NEET 2021, I guess, right? And even after that also that question was repeated, right? That hollow organs of our body, right? Hollow organs of our body, they are lined with the smooth muscle fibers. Hollow organs of our body, they are lined with the smooth muscle fibers, because the smooth muscle fiber, it undergoes rhythmic contraction and relaxation. It undergoes rhythmic contraction and relaxation. So rhythmic contraction, relaxation, contraction, relaxation. So you know the food that we eat, how that food will pass, right? The food that we are eating, how will it go to the stomach? Peristalsis will be there. How will it pass through the alimentary canal? Peristalsis will be there. Rhythmic contraction, relaxation, contraction, relaxation, contraction, relaxation. In the uterus also, right? In the uterus also. So of course during pregnancy, you can say that the smooth muscle will be the largest smooth muscle then. So we know that in the case of uterus, endometrium is there, innermost glandular layer. Then the middle one is what? Middle one is myometrium. Middle layer. Jaise outermost is perimetrium. Let me write it. It is perimetrium. Middle one is myometrium. Myo word is associated with muscle, right? So myology. What's myology? Myology is the study of muscles. What's myology? Myology is the study of muscles, right? It is what? It is the study of muscles, right bache? Right bache? And the innermost is endometrium. Innermost is what? Innermost is endometrium. Okay, so this is myometrium. Myo, M for middle, or M for muscles. So here also smooth muscles are there. So during the parturition, during the childbirth, you know na, during the parturition, during the childbirth, right, hypothalamus will give the signal to posterior lobe. Hypothalamus has formed the oxytocin, but it is stored there in posterior lobe. Posterior lobe will release the oxytocin. So right now I'm not talking about the fetal ejection reflex, this and that. Simply I'm saying that oxytocin will act on these muscles and they will undergo rhythmic contraction and relaxation. That also facilitates the childbirth. That's the point that you need to remember. Is that clear? Is that clear? Now let's talk about the cardiac muscle. From the word itself, you can understand that they are associated with our heart, right? What is our heart? It is the hollow muscular organ. What is the heart? It is the hollow muscular organ made up of muscles. It is hollow, it is muscular. Okay, so cardiac muscles are associated with it. They are also having the striations. They are also having the striations. So they are in the wall of heart, bache. They are also cylindrical in shape, uninucleated, but here you will see the fibers are branched. So sometimes we miss such kind of details, which is not right. We should know that they both are unbranched, but cardiac muscles, they are branched. Now all the cardiac muscles, they need to work together as a unit. They form a single unit, right? They form a single unit, so they are branched as well. So in the case of cardiac muscles, again that's a PYQ. This point is very important, that they have intercalated discs. Do check the previous year questions. You will get this question. Intercalated discs, they are present in. Intercalated discs, they are present in. They are present in the cardiac muscle fibers, right? They are, where are they present? They are present in the cardiac muscle fibers. What? Intercalated discs. Are you getting my point? Are you getting my point? So it's like, let's say you have the branches are there. Why they are branched? Because they have to work together as a single unit. Heart will contract and relax, we know it very well. So when that wave of contraction will pass, all the muscle fibers, they'll contract simultaneously, they'll work together as a unit. So in between that continuous connection is your intercalated discs, right? That is your intercalated disc. So when you talk about the heart muscles, remember, they never get fatigued. They are also involuntary in action. They are also involuntary in action. Understood? Take the screenshot, please. Take the screenshot, please. Right? Right? Okay. So intercalated discs, where are they present? They are present in the cardiac muscles. They will connect all the muscles together. So they are the example of the cell junctions, right? They are the examples of the cell junctions. So do not forget about it. Okay. So skeletal muscles, cardiac muscles, branching is there, right bache? Striations are also there, and they have intercalated discs. What do they have? They have intercalated discs, right bache? Which is a kind of junction in between these muscle fibers. Okay, right? So action potential will pass through it very quickly, right? So it's a kind of gap junction here. It's a kind of gap junction here. Baki ye dekho, smooth muscles, right? So visceral single unit smooth muscles, visceral multi-unit smooth muscle tissue, spindle shaped they are, right? So that's what it is. This is all about the muscles here. Now quickly revise it from the NCERT, and then, my dear students, we will discuss the ultrastructure of muscle. So if the ultrastructure of muscle is clear, then trust me, the sliding filament theory will also be very easy, right? Then the sliding filament theory will also, it will be easy for you all. Okay, right? So is it fine? So up to this part, any doubt? Up to this part, any doubt? I hope the speed is also okay. So it's a quick one shot. We have to cover the syllabus. We have to cover all the important points here. So tell me, any doubt, or is it all clear? Any doubt, or is it all clear? Yes, please. The speed is fine. The speed is fine, right? Okay, so visceral muscles means you are talking about the smooth muscles here, right? You are talking about the smooth muscles here. As I said, they are located in the inner walls of hollow visceral organs, right? PYQ. It's a direct PYQ. In the alimentary canal and reproductive tract it is, right? They don't have striation. That's why the word smooth is. And as I said, they are fusiform, right? They are spindle shaped, okay? So they are not under voluntary control of nervous system. Matlab they are involuntary. We cannot control it with our own will. Like if I want to move my hand like this, I can, because skeletal muscles are associated with it. I can control them with my own will, but I cannot control smooth muscles and the heart muscles. Okay. So in transportation of food through digestive tract, the movement of gametes, for all that, these muscles are going to help. Then bache, cardiac muscles, right? They assemble in a branching pattern. They form the cardiac muscles. So they are also striated. They are also involuntary. Highlight the important words here, but we cannot control them with their own will. And what do we have there? Intercalated discs are present. That's important. That's very, very, very important. Okay, that's very, very, very important. Now, so each organized skeletal muscle in our body, each organized skeletal muscle in our body is made up of number of muscle bundles, right? It is made up of number of muscle bundles. It is made up of number of muscle bundles or fascicles. Chalo. Now let's discuss the ultrastructure of muscles. Okay. Ready? Then we'll revise it from NCERT. So muscles are mesodermal in origin. Muscles are mesodermal in origin. Please repeat it. Muscles are, right, mesodermal in origin. Please repeat it. Yes, kindly repeat it. Muscles are mesodermal in origin. Yes, please. So let's say if this is the muscle that we have. Now, bache, when you talk about the muscle, we'll discuss the layers, we will discuss everything, but please pay attention here. Okay, they are mesodermal in origin mainly. This is the muscle that we have. Muscle is having many muscle bundles. Okay, in the muscles, what will you see? You will see muscle bundles. Many muscle bundles together, they form the muscle. Right, when you talk about the muscle, many muscle bundles are going to, together they are going to form the muscle. Just for an example, side view is something like this, front view is something like this. Like when you are talking about a muscle, you will see many muscle bundles there. What will you see there, bache? You will see many muscle bundles there. It's like one muscle bundle, right? This is the second muscle bundle, right? This is the third muscle bundle. Okay, this is the fourth. I'm not specifying the number here. I want to tell you is that so many muscle bundles together, they form the muscle, right? So many muscle bundles together, they form the muscle. This is what I'm saying. Firstly, understand the terminology, then one by one, I will make everything clear to you, right? So in the muscle, what do we have? We have the muscle bundles. What do we have, students? We have the muscle bundles. We also call them as fascicles. What do we call them? We call them as fascicles. Write down this word here in the chat section, right? That each, so we are talking about the ultrastructure of muscle, and which muscle are we talking about here? We are talking about the skeletal muscles, right? What are we talking here? What are we discussing here? We are talking about the skeletal muscle here, right? We are talking about the skeletal muscle here. Is that clear? Is that clear? Yes. Right? So let me write it here so that you do not miss it. Right? So we have the muscle. So now this is the muscle, and obviously connective tissue, right? Connective tissue will also cover this muscle. Matlab, connective tissue is required now. It is required. Connective tissue is important thing. So basically the muscle itself, right? The muscle is covered by a layer of connective tissue. I'm writing it here. Muscle is covered by a layer of connective tissue. And that is known as, anybody here in the class? That is known as epimysium. That is known as epimysium. Right, bache? It is the layer of connective tissue. But what is the most important thing here? You have to understand that muscle itself, a skeletal muscle is having the it is having the muscle bundles. Muscle bundles are organized there. Right, bache? Muscle bundles are organized there. Muscle bundles are organized there. So the way I said that muscle is covered with the connective tissue sheath, with the connective tissue, I will not use the word sheath here. So it is covered with the connective tissue. We are calling it as epimysium, right? That's what I just said now. We are calling it as epimysium. So same way, when you talk about these muscle bundles are also covered with connective tissue. I'm talking about each muscle bundle here. Each muscle bundle is also covered with the connective tissue, and we call it as perimysium. What do we call it? We call it as perimysium. What do we call it? We call it as perimysium. So again, in a muscle, the organized muscle bundles will be there. We use the word fascicles for them. So all these muscle bundles, first of all, each muscle bundle is covered with a layer, that connective tissue layer, that is perimysium. But now I'm saying that all the muscle bundles, they are connected to each other. They are connected to each other. All these muscle bundles are also connected to each other, right? They are also connected to each other. So for that, we use the word fascia. Right, for that we use the word fascia. Just say for an example, let's say these are the muscle bundles here. So all these muscle bundles are connected to each other. All these muscle bundles are connected to each other. So what is the word that we use for this connective tissue? Here the word is fascia. What is the word, students? The word is fascia. So it is the tough connective tissue layer that is holding these muscle bundles together, right? Right, it is the tough collagenous. It is made up of collagen. You know na, collagen, the most abundant protein of animal world. So tough collagenous connective tissue layer that is holding all the muscle bundles together. together. Okay, so this is fascia. So perimysium is just the connective tissue layer which is covering each muscle bundle. So do not get confused. Now, when you talk about these muscle bundles, in each muscle bundles, what will you see? You will see the anatomical unit of muscle. What will you see here? You will see the anatomical unit of muscle, that is known as muscle fiber. So in each muscle bundle, you will see that muscle fibers are present. What is present, bache? Muscle fibers are present, right? Muscle fibers are present. Please write down. Please write down. So muscle bundles are there. So each, yes. So it contains what? It contains the muscle fibers. It contains what? It contains the muscle fiber. Is that clear? Is that clear? So you can see it like this. Like let's say if this is a muscle bundle. So in each muscle bundle, what do you have? You have the muscle fiber. What do you have here? You have the muscle fiber. So when we talk about this muscle fiber, so bache, this muscle fiber individually, it is also having a connective tissue layer that is covering it. For that we use the word endomysium, right? For that we use the word endomysium. But these perimysium, epimysium, endomysium is not given in NCERT, right? These words are not written in NCERT, right? But yes, obviously they do exist, right? They do exist. Okay, okay. Yes, is that clear? Is that clear? Sure. Is that clear? So they are having the muscle fiber. So when you talk about the muscle fiber, right? Let's say for an example, if this is the muscle fiber, so this muscle fiber is considered as the anatomical unit of muscle. What do we call it? It is known as the anatomical unit of muscle. Anatomical unit of muscle. And now here this muscle fiber, its outer covering, its plasma membrane, right? Its plasma membrane is known as sarcolemma, beta. What is it? It is known as sarcolemma. Sarcolemma. Lemma means layer. Lemma means covering. Right here, this cytoplasm here is known as sarcoplasm. And in the muscle fibers, of course, we know that endoplasmic reticulum is there. That will be sarcoplasmic reticulum, right? That will be sarcoplasmic reticulum. So if it is sarcoplasmic reticulum, remember it's a storehouse of calcium. It's a storehouse of calcium. It's a storehouse of calcium. So can I say that it is basically smooth endoplasmic reticulum? Yes, because it's a storehouse of calcium, and this calcium is very important for the muscle contraction. So understand the structure here. In the muscle bundle, there are muscle fibers. The outer layer of muscle fiber, the plasma membrane of muscle fiber is sarcolemma. The cytoplasm here is the sarcoplasm. The endoplasmic reticulum here is the sarcoplasmic reticulum. And you are talking about the muscle fiber. So it is what? It is a syncytium. What is the meaning of syncytium? It is multinucleated, right? It is multinucleated. Guys, I hope you remember that cynocytic is also the multinucleated condition, but there is a difference. There is a difference in syncytium and cynocytic. Both, like they both are telling us about the multinucleated condition, but still there is a difference. Do check it up. Do check that. Fine. Do check that. Is that clear? Is that clear? Yes. So in the muscle fiber, many nuclei are there. So we are using the word syncytium here. Sarcoplasm is there. Sarcoplasmic reticulum is there. Abundant mitochondria are also present. And mitochondria here is known as sarcosome. What do we call it? We call it as sarcosome. What do we call it? We call it as sarcosome. Any doubt? Any doubt? Any doubt? So remember here in this, yes, in this muscle fiber also, here in this sarcoplasm also, you will see some parallelly placed some proteins. We call them as myofibrils. What do we call them? We call them as myofibril. So it's a protein basically, right? Proteinaceous structure it is. So when you look at this myofibril, here you see dark and light bands due to which striations are there. So this is what we need to understand. This is what we need to understand now. Is that clear? This is what we need to understand now. We have to understand, we need to understand more about the muscle fiber as well. So let's have a quick revision. Let's have a quick revision. So basically we are talking about the muscle. We are saying that these muscles are well organized structure, they are having the muscle bundles. What do we call these muscle bundles as? We call them as fascicles. Now we are saying that the connective tissue that is holding all these muscle bundles together is known as fascia. What is another name for muscle bundles? It is fascicles, right? So do not get confused. They are fascicles. Muscle bundles are fascicles, and the connective tissue layer which is holding it all together, it is known as fascia, right? It is known as fascia. And now when you look at this part here, when you look at these muscle bundles here you will see within these muscle bundles, muscle fibers are present. What is present? Muscle fibers are present, and this muscle fiber is the anatomical unit of muscle, and here its outer covering is sarcolemma, sarcoplasmic reticulum, sarcoplasm, sarcosome is the word that we use, and here in the muscle fiber it is syncytium, multinucleated condition is there, and you will see these parallelly arranged protein fibrils. We call them as myofibrils. What do we call them? We call them as myofibrils. We call them as myofibrils. Is that clear? Is that clear? Yes, please. Is that clear? So you can see the language here. So when we discuss the ultrastructure of skeletal muscle, right? So each organized skeletal muscle is made up of number of muscle bundles. So the word here is fascicle. So the another name is important. Who's going to hold it together? Common collagenous connective tissue, fascia. So each muscle bundle is having number of muscle fibers, right? It is having number of muscle fibers. Understood? Number of muscle fibers. Understood? Chalo, I'll come to this diagram later. Right? So the layer here is sarcolemma enclosing the sarcoplasm. So muscle fiber is a syncytium, means multinucleated. It is. Right? Right. So sarcoplasmic reticulum, that is SR. It is the storehouse of calcium ion. So it is the smooth endoplasmic reticulum. It is a storehouse of calcium ion, and these calcium ions, they are very important for the muscle contractions, and that is what we need to understand. Okay, bache? So characteristic feature of muscle fiber is the presence of large number of parallelly arranged filaments, and that is the myofilaments or myofibrils. What do we call it? That is the myofilament or myofibrils. What do we call it? It is the myofilaments or myofibrils. Is that clear? Is that clear? So now let's discuss the structure of this myofibril. Right, bache? Let's discuss the structure of this myofibril. Yes. Do you know about this structure? So please pay attention, because of course we are going to understand the structure of the sarcomere, and it is important. Right? So structure is clear, matlab everything will be easy. Okay? Okay? And any doubt? Any doubt? Any doubt? No? Very good. So we are saying that they have alternate, that is what we are saying now, that there are alternate dark and light bands. Right? This is what we are saying, that they have alternate dark and light bands. These myofibrils, what do they have? They have alternate dark and light band. Dark and light band. Dark and light band. Please pay attention. Example, this is the myofibril. You will see striation there. Why? Because there is alternate dark and light band. So when you are saying alternate dark and light band, just for an example, this is the light band, then dark, light, dark, light, dark. That's how it is. Now there are two bands, I band, I, light band, I is present there. We are saying now light band, I is present there. I band it is dark, arc, right? Dark, arc, A is there. So light band and dark band is there. I band and A band is there, right? So A for anisotropic here, I for isotropic here. Firstly, understand this basic thing. We need to discuss the triad, T-tubules, everything, but firstly focus here. Light band, dark band. I. So in this light band, what are you going to see? In this light band, what are you going to see? You will see actin filaments are present. What is present? Actin filaments are present. Which protein is present here? Actin protein, actin filament in I, like this. Dark band, like when we are in our dark phase, our good friends, they don't leave us. That's how I remember that in the dark band you have myosin and actin. So in NCERT only myosin is given, but in the dark band also we will see the area where actin filaments are also there. I will discuss it. Okay, so actin filament and myosin filament. So actin, it is also the thin myofilament. It is also the thin myofilament, and your myosin, it is the thick myofilament. What's that? It is the thick myofilament. What's that? It is the thick myofilament. Is that clear? So alternate dark and light band, alternate dark and light band, alternate dark and light band. Just for an example, this is the I band, A band, I band, A band. Okay. So now from this I band, or you can say that now from this, yes, now from this I band, this I band, it is bisected. Do you know that? I band, it is bisected. So when we are saying it is bisected, it is divided into two parts by this Z line, Z line or Z line. This Z line, what will it do? It will bisect I band. It bisects I band. It bisects I band. What is it? It is the Z line. What is it? It is the Z line. What will it do, bache? It bisects I band. Am I clear here? It bisects I band. So can I say that it divides it into two equal halves? Yes, bache. So this Z line, right? It is basically made up of what? It is made up of elastic fibers. It is made up of elastic fibers. Everything is given in NCERT, right? It is made up of what? It is made up of, right, elastic fiber. It is the elastic fiber which is bisecting it, right? So these filaments, right, these thin filaments, they are very firmly attached to this Z line. These thin filaments, they are very firmly attached to this Z line. Is that clear? Is that clear? Now when you are talking about this A band here also, in the A band also, what will you see? In the A band, you will see that from the center here, from the center, this M line will cross from the center, middle, M line, middle, right? This M line will cross, and this is a thin fibrous membrane. It is the thin fibrous membrane. It is the thin fibrous membrane. Look at the structure here. You are talking about the myofibrin. You are saying alternate dark and light bands are there. We are saying that alternate dark and light bands are there. What is there? Alternate dark and light bands are present here. Okay, alternate dark and light bands are present here. So when you are talking about the I band, it is having thin myofilament, and we are saying that it bisects the I band. Okay, so this Z line is having elastic fibers. Now here you have the thick myofilament. Here you have the thick the dark band here. The dark band here. Here in the center, the M line is there, which is the thin this thin fibrous membrane. Now this part is actually your thick myofilament. It is actually your thick myofilament, right, bache? And it is made up of myosin protein, right? It is made up of myosin protein. And let me tell you that this myosin is also attached here to the Z line with the help of titin protein. With the help of titin protein, right? It is attached here. First of all, tell me this structure clear? This structure clear? First of all, tell me this structure is clear to you or not? I am saying I band, light band, isotropic, dark band, A band, anisotropic. I band will have actin filament, A band will have myosin filament. But here in the A band, at the ends, you will see that how this actin filaments, they are overlapping this myosin now, right? Actin filaments, they are basically overlapping this myosin now in the A band here. So this is the I band. This is the I band. This part here is the A band. This part here is the A band. And when you look at the A band, you will see that here the middle portion, right? Here, what will you see? You will see, students, that the middle portion here, right, this middle part, here only myosin is there. It is not overlapped by actin. Here only myosin is there. It is not overlapped by actin. It is known as H zone. That is Henson zone. It is known as H zone. That is Henson zone. Okay, I'm repeating the points again. What am I saying? I band, A band, I band. Alternate dark light dark band, they are present. In the light band you have actin, in the dark band majorly you have myosin, but the ends here of the myosin are overlapped by actin. You have to consider this point. Now in between here, you know that dark band is having the M line passing through it, which is the thin fibrous membrane. And when we look at this A band, we also see, when we look at this dark band, we also see that this middle wala portion here, myosin is not overlapped by actin. So we call it as H zone. What do we call it? We call it as H zone. H zone, Henson zone it is. Henson zone it is. And these myosin filaments, they are attached to this Z line, and this protein is tightened. This protein is tightened, right? So this is the information that I have delivered to you. Now I am saying that I band, it is bisected by the Z line, which is the elastic fiber. Now the part in between two successive Z line, right? So basically this part of myofibril which is in between, right, this part of myofibril which is in between two successive Z lines, one after another, this is the sarcomere, right? What is it? It is the sarcomere, and it is the functional unit of contraction. What is it, bache? It is the sarcomere. Yes please, it is the sarcomere. Yes please. So it is the functional unit of contraction. It is the part that will undergo, like it will shorten during contraction. So it is the functional unit of contraction. So what do we have in a sarcomere? Look at it. So the portion of myofibril in between two successive Z lines, that is sarcomere, which is the functional unit. So when you look at this sarcomere, what will you get? Two half I band and one A band. Right? If you understand the diagram, then you can relate. What will you see here? Two half I band and one A band. Two half I band and one A band. That's what we have here. So when we talk about the sarcomere, right, when we talk about the sarcomere, so you will see that there will be two half I band plus one dark band. That's what we have. So this sarcomere is what? It is the functional unit of contraction. What is it? It is the functional unit of contraction. What is it? It is the functional unit of contraction. Is that clear? So two half I band, one A band. Two half I band, one A band. Two half I band, one A band is present here. Any doubt? Any doubt? Yes? Any doubt? This is what sarcomere is. This is what sarcomere is. So where these structures are present, let's say this is the muscle fiber. I know the diagram is not up to mark. So see, muscle fiber is having parallel myofibril. So basically we are zooming in that myofibril and we are checking it. Check. So when you talk about this myofibril, when you talk about this muscle fiber, it is the anatomical unit of muscle. It is the anatomical unit of muscle. And this sarcomere is the functional unit of muscle. So this, what is it? This sarcolemma, at places you will see it shows invagination. At places you see it shows invagination. It shows invagination, right? It will form a structure like this. It will form a structure like this. And you see that here in the sarcoplasm, the sarcoplasmic reticulum is around this invaginated structure. It's like that. So in the structure of muscle fibers, that is what we see, that sarcolemma at places, it forms the invagination. So we call it as T tubules. We call it as T tubules. And in their surrounding, like on their either sides, you will see sarcoplasmic reticulum. So this part, like T tubule, this invagination, and the sarcoplasmic reticulum here, we use the word triad for it. Right? We use the word triad for this. So you know that when there is the muscle contraction, when there is the stimulus for muscle contraction, you know that neuron will bring it. right? Right? Motor neuron will come, है ना? And it will tell the muscles that now it's the time for the contraction. So when you look at the endings of this motor neuron and the muscle, the junction here in between is bache neuromuscular junction. The junction here is in between bache neuromuscular junction, and we also call it as motor end plate or motor plate. We also call it as motor end plate or motor plate. So the signal will come, the neurotransmitter will release here, so it will pass that stimulus to the muscle. So through this T tubules, it will tell the sarcoplasmic reticulum that please release your calcium ions. So when the calcium ions will be there in the sarcoplasm, this muscle contraction will start. This muscle contraction will start. So this is all about the structure here. Now we will understand it with the help of diagrams. How are we going to understand it? We will understand it with the help of diagrams. Okay, bache? Okay. And one more thing that you need to remember, that muscles, they are attached to bones with the help of tendons. With the help of tendons. So tendon is an example of connective tissue. It is an example of connective tissue. It is an example of connective tissue. So, this is the diagram. So this is your bone. Muscles are attached to it with the help of tendon. Okay, so muscle is having muscle bundle. This muscle is having what? It is having the muscle bundles. It is having the muscle bundles. So these muscle bundles are having what? I told you now, this muscle bundle. So all these muscle bundles are connected together with the help of fascia. So each muscle bundle is fascicle. You can see the slides here also, right? Now here in this muscle bundle, what will you see? You will see the muscle fiber, right? The anatomical unit of muscle, which is covered with the connective tissue endomysium. So here in between, you will see the parallelly arranged myofibrils are there. At places you will see the T tubules also, right bache? So this is the myofibril having alternate dark and light band, and here the structure in between two successive Z line is the sarcomere, right? It is the sarcomere. So please look at this diagram carefully, right? Then I will move to the next part. Okay, so see this diagram, muscle, bundles, right? The muscle fiber, the myofibrils, and this is the myosin, thick filament. Okay, doubt, Henson zone and all clear? Diagrams are very important, and I told you what epimysium, what perimysium, and what endomysium is. So you can see this diagram, that sarcolemma at places it is forming the invagination. You can see this, that is the T tubule, and on its both the sides sarcoplasmic reticulum is there, so we are calling it as triad. Look at the structure, look at the diagram, please look at the diagram. Right? You understand the diagram. I'm not saying you have to memorize it, understand it. When you understand the structure of muscle, when you understand everything, then it will be easy for you. Right? Just ye dekho, these mitochondria are made in between. Right? Look at the structures there. Look at the structures there. Done, bache? So this is the diagram in NCERT. Right? So blood capillaries and all, they are supplying this. They are here, and this is the structure. Yes, this is the structure. So now the next thing that we need to understand is, ye dekho, and this is the diagram which is given in NCERT, the Z line, A band. So this is the H zone where myosin is not overlapped by actin, where myosin is not overlapped by actin. So the portion between two successive Z line is the sarcomere. Okay, it is the sarcomere. Clear? It is the sarcomere. Again, observe the diagram, right? Observe the diagram. Okay, this is a titin protein. With the help of it, this myosin is attached to the Z line. Diagram is important, kindly observe it. So ye dekho, this is a relaxed muscle. When the muscle is contracted, sarcomere size will decrease. Alright? I'll explain it. You check the diagram first. You check the diagram first. Right? So if the diagram is clear na bache, trust me, if the diagram is clear na, everything will be easy. And trust me, even the session is not going to be lengthy. Within 4 hours we will finish it. 3 hours or 3.5 hours we will finish it. Okay, chaliye. So look at the diagram. Okay, and then we will continue. Just allow me 1 minute. Ma'am, please explain properly. Yeah, I have explained properly. I have explained each and every line here. Okay, you can just watch the lecture again. Don't worry about it. So again, I'm saying, students, if the diagram is clear, everything is clear. I am here only. Just watch this, right? Just carefully observe this. Just carefully observe this and tell me if there is any doubt. If there is any doubt, the diagram should be clear. Yes, please. The diagram should be clear. Done? Is it done? Clear? So if it is a yes, some fire emojis in the chat section. If it is a yes, then some fire emojis in the chat section. Okay, so now we will discuss the structure of the contractile proteins. Contractile proteins which will undergo contraction. What are we talking here? What are we discussing here? We are talking about the myosin and actin. What are we talking here? We are talking about the myosin and actin. So let's discuss this part again, right? That what the structure of contractile protein. So of course we will start with the actin, right? We will start with the actin. We will start with the thin myofilament. We can start with thin and thick both. Chalo, let's start with the thick myofilament. So when we are saying thick myofilament, when we are saying thick myofilament, what are we talking? We are talking about the myosin structure here. What are we talking? We are talking about the myosin structure here. So let's discuss it. Okay, let's discuss it. Myosin. Dekho, it is made up of, like, it's a polymeric protein, right? Polymerization will result in the formation of this. In simple words, firstly, I'll explain that your meromyosins, right, they are the monomers. Firstly, in very simple words, I will explain that your meromyosins, right, they are the monomers, but they will polymerize. So many meromyosins together will form your myosin, this thick filament. Right, they will form your thick filament. Just say, what will you see here? When you look at the structure of meromyosin, it is something like this. It is something like this. It is even given in the NCERT. It is something like this. See, again I'm repeating. When you are talking about the myosin filament, it is the thick filament. It is a result of polymerization. What is going to polymerize? The monomers. So when you are talking about the monomers, you are talking about the meromyosin here. What are you talking? You are talking about the meromyosin here. And when we discuss about these meromyosin, like something like this is given in our book, we have to discuss this part. So many, firstly understand that so many meromyosins together, right? So many meromyosins together, they will polymerize. Yes, students, so many meromyosins together, they will polymerize, and they are the one that will give us this thick filament. That will give us this thick filament. So we need to understand what this meromyosin is. So when you talk about the meromyosin, you divide this meromyosin structure into two parts, right? You will divide it into two parts. You will call it as heavy meromyosin and light meromyosin. Heavy meromyosin and light meromyosin, right? That is HMM and that is LMM. That is HMM. That is LMM. Right? What's that? It is HMM. It is LMM. Heavy meromyosin and light meromyosin. So basically, we are going to discuss this particular part. Okay? Okay? Okay, so when it is the heavy meromyosin, what will you see here? A globular head with short arm. A globular head with short arm. What will we see here? A globular head with short arm. And here you will see the tail. Here you will see the tail. So basically, globular head with short arm. Globular head with short arm and the tail. Just say, let's suppose it is the tail. This is the globular head. Right, and let's say this part is a short arm. So it is forming your heavy meromyosin and this tail is forming your light meromyosin. It is forming your light meromyosin. Now, in the heavy meromyosin, when we are saying a globular head, this globular head is having actin binding site. Yes, bache. Actin binding site, matlab this head will bind with the actin, and that is important for sliding filament theory. That is important for muscle contraction. Plus, it is also having the ATP binding site, right? It is also having the ATP binding site. So basically, in this globular head, ATP binding site and actin binding sites are there. So this globular head is an active ATPase. It will hydrolyze this ATP. It will hydrolyze this ATP. That is again a previous year question that you need to remember. right? It is a previous year question that you need to remember. Are you getting my point? It is a previous year question that HMM, that is heavy meromyosin, it is having the globular head. That head is having actin binding site and ATP binding site. So basically this globular head is an active ATPase, it hydrolyzes the ATP. And when you are talking about the light meromyosin, it is LMM, right? It is LMM, right? This is the tail region here. So tail is not the active ATPase, it is the head region. Firstly, keep it in your mind. Right, firstly keep it in your mind. Now come to the NCERT, right? Each and every point is written here in NCERT. We just need to decode the NCERT. So we will start with this portion first, that each myosin filament is a polymerized protein. Why are they saying also? Because they here, firstly, they have discussed the actin. We will discuss it later. Then many monomeric protein, meromyosins, they will form one thick filament. These meromyosins, they will polymerize, they will form your thick myofilament. Now, each meromyosin is having two important parts, a globular head with short arm and a tail. So globular head with short arm, the former is HMM and the later is LMM. So HMM, it is having the head and short arm. It projects outwards at regular distance and angle from each other from the surface of polymerized myosin. Matlab, what is happening? Just let's say, when these meromyosins, when they will polymerize, these meromyosins, when they polymerize, so at a regular interval, the gap is also regular and at a regular proper angle, dekho, they will project outwards. Just this head and this short arm, this will project outward like this. It will project outward like this. It will project outward like this. So it is projecting. Just let's say next is this. So they will project outward, right? They will project outward at regular distance. Just let's say if here this is the distance, next distance will be like this. At a regular distance, they will project out, right? They will project out and regular distance and an angle, right? Just this is the angle, this is the angle. They will project out from the surface of polymerized myosin, right? So look at this. At regular distance and an angle, they will project out from the surface of polymerized protein like this, right? So this structure is known as the cross arm. What is the word that we use for it? We call it as cross arm. So globular head is an active ATPase enzyme. It hydrolyzes the ATP. It has binding sites for ATP and the actin, already discussed, right? Already discussed. Any doubt? That's the diagram. So actin binding sites here. In the paper, they can ask you to name it, right? Diagram based questions are also there now. This is the ATP binding site. So actin binding site is there, ATP binding site is there. Actin binding site is there, ATP binding site is there. So this is your LMM, light meromyosin, head and short arm, right? At regular intervals they are projecting. We are calling it as cross arm. So this is your HMM, that is heavy meromyosin. Any doubt in the diagram? Any doubt here in the diagram? Yes, please. Any doubt here in the diagram? Clear? Now let's talk about the actin filament, because that is also the result of polymerization only. So actin filament, that's the word. Actin filament, that's the word. So it is the thin filament. It is the thin myofilament. What is it, bache? It is the thin myofilament. Is that clear? Is that clear? Yes. Is that clear? So it is the thin myofilament, actin, right? So here we will discuss about three proteins. We will talk about actin, we will talk about tropomyosin, and we are also going to talk about troponin. So basically in your thin myofilament, three proteins will be there. We will talk about the actin, tropomyosin and troponin. So let's start with the actin here. So what's going to happen? The polymerization again, right? The polymerization again. Okay, so actin is what? Bache, it is a globular protein. It is a globular protein. Again, everything is written in NCERT. Let's try to understand that first. Right, let's try to understand that first. Right, so it is a globular protein and has low molecular weight. And what are we going to call it? We will call it as G-actin. What will we call it? G-actin, globular actin. It is monomeric one. It is having the low molecular weight. Just for an example, this is your G-actin. It is also having the binding site for myosin. This is your G-actin. So what's going to happen? Your G-actin, it will polymerize. It will polymerize. Of course, magnesium ions are required for it. So your G-actin, what will happen to your G-actin? It will polymerize, 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 polymerize. Your G-actin will do what? It will polymerize. Your G-actin will do what? Yes, bachya bhin hai. It will polymerize. And it will result in the formation of F-actin. So F-actin is polymerized one. It is the filamentous or fibrous one. Right. So globular and filamentous. Now what will happen? Basically, two filaments of actin, they'll be helically arranged. Right? Just let's say one filament, so two F-actins. First of all, G-actin, it will polymerize to form F-actin. G-actin is monomeric, F-actin is polymeric. G-actin is monomeric, F-actin is polymeric. G-actin, it is monomeric, F-actin, it is polymeric. It is polymeric, and that F-actin will polymerize. That F-actin will polymerize. Are you getting my word? Two F-actins, they'll be helically arranged. So G-actin monomeric, it will polymerize, it will form F-actin. So these two F-actins, two F filaments, they will be helically arranged like this. They'll be helically arranged like this. And remember, they will have the binding site for myosin. Just a myosin head, it will attach to the actin. Na? So actin should also have the site to which that myosin can bind. So yes, this G-actin will have that site, and that's how it is. So this part clear? Firstly, tell me this part clear. Everything is written in NCERT. You understand the concept. Okay. Now here, when you talk about this helically arranged F-actin filaments, you will see one more protein, right, is also present around it. This is your tropomyosin. It will also cover it throughout the length. This tropomyosin will also cover it throughout the length. Just say, for an example, you have a book, right? You have a book, you know that it is having an outer cover. You are again putting, right? You are again putting a brown paper on it. Just when we go to the school, we have the notebooks now. So we have the notebooks, we put the covers on that brown paper, then we put the sticker. This is the chemistry notebook, physics notebook, biology notebook, like this, like this. So same is the case here. So G-actin will polymerize to form F-actin. Magnesium ions are also required here. Then these two F-actins, they'll be helically coiled, right? They will be helically coiled. They'll be helically arranged. And these two F-actins, they are covered with what? These two F-actins, they are, throughout their length, they are also covered with the tropomyosin. So this colored protein here is the tropomyosin. So remember, this tropomyosin, what will it do? This tropomyosin, what will it do? It is covering the F-actin is in such a way that it will cover the sites on F-actin that can bind with the myosin. Right, that can bind with the myosin. It's like that. It's like that. And here at places you will see. Here at places you will see a trimeric protein. Troponin is also present. A trimeric protein. Troponin is also present. A trimeric protein. Troponin is also present. So rest everything is written in NCERT. About the troponin, it is not given there. Like, so I'll add some extra points related to the troponin here, then we'll just read the NCERT. So when you talk about the troponin, it's a complex protein. It's a complex protein. Understand the structure, everything will be easy. Complex protein made up of 3 polypeptides. It's a complex protein made up of 3 polypeptides. Okay, it is made up of 3 polypeptides, and this troponin is distributed at regular intervals on tropomyosin. This part clear that it is a trimeric protein made up of 3 polypeptide chains, right? So here there is troponin tick, TIC. That's how you can remember. Just let's say if we are showing it like this, right? If we are showing it like this, that it is troponin, 3 polypeptides will make it. So it is TIC, troponin tick, troponin tick. So troponin T, it is attached to, of course, right? It is attached to the tropomyosin, right? There are 3, tick here. So T is attached to the tropomyosin, right? Yes, troponin T, it is attached to tropomyosin and it is attached to troponin tick it is na. So it is attached to, once again, it is attached to tropomyosin and to two other components of troponin. So it is troponin T. I am telling you 3 polypeptides are making this trimeric protein, troponin T. Obviously, it is attached to the tropomyosin plus to 2 other components of troponin. What about E? I, troponin I. I means inhibit. So this part, what will it do? It inhibits actin and myosin interaction. It will not allow the actin and myosin to interact, and obviously it is also attached to the 2 other components of troponin. So troponin I inhibits the F-actin myosin interaction, and obviously it is also attached to 2 other components of troponin. That is so obvious now. That is obvious. Now, the third thing, right? So troponin T clear, troponin I clear, and then it is the troponin C. Then what is it? It is the troponin C. So C for calcium. So it is attached to the calcium. It will attach to the calcium ion at the time of muscle contraction. Plus, it is also attached to the 2 other components of troponin. So if I have to show you, like if I have to simplify this, like let's say if it is troponin T, it is I, it is C. So just say, let's say it is attached to the tropomyosin as well as to the 2 other components of troponin. This I, it has to stop the interaction of actin and myosin. It is also attached to the 2 other components. This C, it will attach to the calcium ion at the time of muscle contraction. It is also attached to the 2 other components. So as I said, if the diagram clear, trust me, all other things will be easy. Okay, this is C. In the NCERT, what they have given, they are saying that each actin filament is made up of 2 filamentous actin helically bound to each other. How that F-actins will form? F-actin is a polymer of monomeric G-actin. So 2 filaments of another protein, tropomyosin, also run close to the F-actin throughout its length. You know now, 2 filaments of tropomyosin, they are also running right close to this close to this F-actin, and it has basically covered the sites on actin which can bind with the myosin. Okay, so troponin is also distributed at regular intervals on tropomyosin. So in the resting state of a subunit of troponin masks the active binding site of myosin on the actin filament. Give it a reading quick. So this is all about the structure of the filaments here. Yes, give it a reading. Done? Sure. That's the diagram, and look at the structure here. G-actin is forming the effectin. These are the sites that will bind with the myosin head. So tropomyosin is also covering it throughout the length, right? So troponin is also present, right, bache? So now we have to discuss the mechanism of muscle contraction. But as I said before that, the thing that should be clear is that the structure here. If the diagrams are clear, trust me, rest everything is going to be easy. So now we will start the mechanism of muscle contraction and the sliding filament theory. The sliding is there. So sliding filament theory explains this mechanism of muscle contraction. It is the sliding filament theory that explains the mechanism of muscle contraction. Yes, please, any doubt? Any doubt here? Any doubt here? If there is any doubt, do let me know. So you know that muscles, so major components of muscle. Major component of muscle is water. What is there in the muscles? It is the water. Major component is water. And potassium ion is the most abundant. Do you know that? It is the potassium ion which is the most abundant mineral element of the muscles. Right, bache? It is the most abundant mineral element of muscles. Right? Of muscles. So what am I saying? That the major component of muscle is water and potassium ion is the most abundant mineral element. Other mineral elements, sodium, calcium, phosphorus, magnesium, they all are present. Baki, you know that muscles, if we talk about the muscles, they can store glycogen, you know na? They can also store glycogen. Do you know that? Yes. Right, so they store glycogen as a reserve food material, and in the muscles we have the pigment myoglobin, which is a tertiary protein, and this myoglobin, it can store the oxygen. Just hemoglobin helps, the hemoglobin, that is the quaternary protein, it helps in the transport of oxygen, but this myoglobin here, it helps in the storage of oxygen. It stores the oxygen. Okay, bache? Right, bache? Right, bache? So this is the oxygen carrying red pigment myoglobin, right? So muscles may, other than that, phosphocreatine, creatine, urea, right? All these things are present. This is about the muscle composition. This is about the muscle composition. And already I told you, see, I'm telling you about the sliding filament theory. Obviously, muscles, when they will get the signal for the contraction, only then they will contract. Only then they will contract. Just as sensory neurons. What's the trick here, students? The trick here is ASMI. ASMI. ASMI means efferent neurons. They are sensory. They are also known as sensory neurons. They are the one that will carry, right? They are the one that will carry that information to the brain, right? Now, through motor neurons, which is the efferent, also known as efferent neurons, right? So motor neurons will give that processed information, like from the target organ. The information will be carried by sensory neurons to the central nervous system, to the brain, right? And motor neurons will bring back that, right? That this should be the response to the target organ with the help of motor neurons. So this is a motor neuron, this is the muscle. So the junction in between is known as neuromuscular junction or motor end plate or motor end it is. That's the word that we use. So obviously when it will receive the impulse, right, from dendrites, it will move towards the cyton cell body, and then the axon will carry it away from the cell body. It will reach to the axon terminals here, right? You know that the synaptic vesicles, the neurotransmitters are present here in synaptic vesicles. They'll release the action potential will pass to the next cell. So basically this action potential will pass to the muscle. So this is the neuromuscular junction here, right? This is the neuromuscular junction here. What is it? It is the neuromuscular junction, the junction between motor neuron ending and the muscle fiber, right? It is the junction in between motor neuron ending and the muscle fiber, remember, and the muscle fiber. So this is the junction in between the motor neuron ending and the muscle fiber, which is the neuromuscular junction, my dear students. Motor end plate it is. So it is the specialized portion of sarcolemma of muscle fiber which is in contact with nerve fiber. Is that clear? Now we have to understand the mechanism, which is best explained by sliding filament theory. And who gave that sliding filament theory? Henson and Huxley. Who gave that sliding filament theory? Who gave that sliding filament theory? Henson and Huxley. Do check the spellings here, right? So Henson and Huxley, they gave the sliding filament theory. Okay, bache. So mechanism of muscle contraction is best explained by sliding filament theory, which states that the contraction of muscle fiber take place by sliding of. This line is important. Please pay attention. It is done by sliding of. It is done by sliding of thin filament over thick filament. That's important. Sliding of thin filaments. You recall the structure of sarcomere. Thin filament, it is also overlapping the myosin filament, right, in the dark band. So what is going to happen? There will be sliding of that thin filament over thick filament. There will be the sliding of that thin filament over thick filament. What will be there? There will be the sliding of that thin filament over thick filament. What is it? What is it? That is basically the sliding filament theory, that thin filaments will slide over the thick filament. So if it is written like this, कि thick filaments will slide over the thin filament, if it is written like this, thick filament will slide over the thin filament, then it is incorrect. Okay, so read the question carefully, right? Read the question carefully. Okay, so yes, you can get this question in the final paper. Okay, you can get this question in the final paper. So make sure you read the question carefully. This is important. Chale. Now, see, during muscle contraction, these are the things that are going to happen. You take the screenshot, then I'll explain. You take the screenshot, then I'll explain. These are the things which will happen during muscle contraction, and trust me, all your questions will revolve around it. Right? These points, if you will remember, then everything will be easy for you. I'll explain it as well. You take the screenshot first. You take the screenshot first. Yes? Okay, chaliye, let's discuss that. See, what's going to happen, you understand. Neurotransmitter, they will send the signal, right? They will send the signal. Firstly, I'll explain it. I'll explain it with the help of diagrams as well. You pay attention. I'll finish this part first, right, regarding the muscles. We will discuss everything. Then I'll give you a short break of 10 minutes and then we will start the skeletal system. So this chapter is not very lengthy, but yeah, it is one of the most important chapters. Okay, so we have to finish it. Now, you listen to the steps here, and still if there is any doubt, you're free to ask the questions, but do respond here in the chat section. So what's going to happen, right? The neurotransmitters, they will pass that potential, action potential to muscle fiber. You know that T tubules are there, that action potential will come here in the surrounding. Sarcoplasmic reticulum is there. So sarcoplasmic reticulum is the storehouse of calcium. So once it will receive that action potential, right, it knows that it has to contract. Calcium ions will release. In very simple words, I'm explaining. Then I will explain it from NCERT. I will explain it with the help of NCERT also. So now when calcium ions are there in the sarcoplasm, what do you think will they do? Of course, this calcium ion will bind to the troponin. And if I have to be more specific, so I will say that it will bind to what? It will bind to troponin C. Try to imagine. You have the diagram. You have seen the diagram. You know what diagram, you know what kind of arrangement it is. So that calcium ion will bind to what? It will bind to troponin C. Now when this calcium ion will bind to troponin C, now there is a kind of sliding movement. Like now troponin is like, wow, I got the calcium. So once it will attach itself to the calcium, the way it will move, it will also, because troponin is also attached to the tropomyosin. We know it. Troponin is also attached to the tropomyosin. We know it. So when the calcium will attach to this troponin, because this troponin is also attached to tropomyosin, it will move the tropomyosin or slide the tropomyosin from that actin filament. Basically, what is the thing that we need here? We already know that F-actin, right, throughout the length of F-actin, the tropomyosin protein is also present. And this tropomyosin protein has masked the myosin binding sites on the actin. So when the calcium ion is released in the sarcoplasm, it will attach to the troponin, right, due to which this troponin will slide, right, due to which even the tropomyosin will slide, or you can say that it will move, it will unmask the myosin binding sites. Just say for an example, if it is like this, your tropomyosin was here, your troponin was here, your troponin, it will attach to the calcium ions, due to which it will move this tropomyosin in such a way. This is just to show you so that you can understand that the masking sites, right, that the myosin binding sites on the actin, they will be exposed. They will be exposed. Now, this myosin head will bind here. How? How will it bind? Now you know that in the case of myosin, right, this head, it is having the actin binding site also, it is having the ATP binding site also, and it is an active ATPase. Now obviously this thing is going to play the role. This thing is going to play the role. Okay, chaliye. So let's read it. That muscle contraction is initiated by the signal sent by central nervous system via motor neuron. You know why motor neuron word is there, right? So motor neuron with the muscle fiber connected to it form the motor unit. It is the junction between motor unit and the sarcolemma of the muscle fiber. Okay? So now when the neural signal reaching this neuromuscular junction, a neurotransmitter will be released. What is the name of neurotransmitter? It is acetylcholine. It is acetylcholine. Right? They have given one example here. It is the acetylcholine that will be released here. Now this acetylcholine will generate an action potential. For that we also use the word depolarization in neural control and coordination chapter. We will discuss it. So it will generate an action potential in the sarcolemma. Right, that just say heart will contract. We use the word systole. We say the heart is depolarized. We say the action potential has generated. So that is how these words are related, right? That is how these words are related. So acetylcholine will generate an action potential in the sarcolemma, means that it will give that signal to the sarcolemma. Now it's the time to contract, right? That signal has passed. So this action potential will spread through the muscle fiber, right, and it causes the release of calcium ion. You know how the calcium ions will release through T tubules that will spread. It will tell the associated sarcoplasmic reticulum to release the calcium ion. So when these calcium levels, they will increase in the sarcoplasm, they will bind with the subunit of troponin. What is the name of that subunit? Just now I told you. What is the name of that subunit of troponin? That is your troponin C, right? That is your troponin C. Okay. Right, and when it will bind to the calcium, it removes the masking of active sites for myosin. For myosin, any doubt? Any doubt? I believe it is clear. Now, utilizing the energy from the ATP hydrolysis. ATP will be hydrolyzed, that energy will be utilized, and that myosin head, that, see, imagine, you know that this is the myosin filament. It is having the head here. You know that the actin filament is at its ends. So myosin head will be like this. It will bind with the actin. How? By the energy utilized by the energy from ATP hydrolysis. So when it will bind to the exposed active site on the actin, it will form a cross bridge. Now look at the diagram here. Look at the diagram here. Right? This is your myosin. This is your actin filament. Right? You know that ends of in the dark band, we have seen that myosin filament, their ends are overlapped by actin. Now that head will bind to the actin by the use of energy by the ATP hydrolysis. That ATP hydrolysis has given the energy. So once it will bind to the actin there, it will slide that actin like this. It will bring that actin close to the center like this. Imagine this is the myosin head. Above all, there is the actin. Imagine like this. So when it is attached, when this head is attached to that actin, we are calling it as cross bridge, and it will be like, chalo, actin filament, come to the center. It will slide it over the center. It will slide it over the center. Are you getting my point? In the Telegram group, in our official Telegram group, I will share one animated video related to it. You see this, you see that, you will feel better. So energy will be from ATP hydrolysis. So see, when to this globular head, ADP and adenosine diphosphate, and this phosphate is attached, matlab energy is there. So first of all, see the ATP attach itself to the globular head. So when, remember this thing, that's a PYQ. When ATP is attached to the globular head now, when ATP is attached to the globular head, then cross bridge will not form. ATP will attach to this globular head, ATP will be hydrolyzed. ATP will be hydrolyzed. That energy is allowing this cross bridge to form. So that's a question. When ATP is attached to globular head, cross bridge will form or not? No, it will not form, because ATP, it has to attach here to this globular head, then it will be hydrolyzed. When it will be hydrolyzed, energy will be released. That energy is helpful for the cross bridge formation. That energy is helpful for the cross bridge formation. And when this cross bridge will form, so obviously this myosin will slide the actin over it. So there is the actin, the thin myofilament will slide over the thick filament. Thin myofilament will slide over the thick filament. Myosin is like that only. Thin myofilament will slide over the actin, will slide over the actin, will slide over the actin, right? And then this ADP and P, when they will leave, right, then again the cross bridge will break. New ATP will, so when they will leave, means new ATP will join. So there is the breaking of cross bridge. There is the breaking of cross bridge when new ATP joins, because firstly it has to hydrolyze, energy will be released. That will help in the cross bridge formation. That's what you need to understand, right? Then there is the sliding. Again, with the help of this diagram, I'm again explaining it. I'm repeating the same thing with the help of this diagram, and then I will tell you. Now imagine now, this is the myofilament, this is the actin. It will bring it, the actin filament will slide, right, will slide over the myosin. We will see that H zone disappear. We will see the length of I band will decrease. So these are the points which we need to take care. Firstly, understand the mechanism. So nerve impulse will arrive at axon terminal of motor neuron. It will trigger the release of acetylcholine. That acetylcholine will diffuse here, right, across the cleft, across this space at here, at motor neuromuscular junction, and this acetylcholine will bind to its receptor in the motor end plate. Here it will bind, it will trigger the muscle action potential. Now, acetylcholine esterase, but this is the enzyme which is present here in this gap, in this cleft, right? It will destroy the acetylcholine. So another action potential does not arise unless more acetylcholine will release. Once acetylcholine will release, it will pass the signal to the muscle will start the contraction. Then of course the acetylcholine present here will be destroyed by the enzymes there. It will be resequestered by this neuron again. Okay. It's like that, right? So once the signal has passed, the action potential has passed, muscle will contract. Then again, new, wave of, basically new impulse will come, then again it will contract. We'll come to that part later. So now what will happen? What will happen? So action potential of muscle, it will travel along the T tubules. It will open the sarcoplasmic reticulum, open the calcium channels on sarcoplasmic reticulum. Calcium will flood here in the sarcoplasm. Now this calcium will bind to the troponin, which is on the thin filament, right? And then it will expose the binding site for myosin. Next step will be, right, because once the sites are exposed, the contraction will occur, the power stroke will be there, ATP will be used, myosin head will bind to the actin, and then it will swivel and it release the thin filament, it pull it towards the center of the sarcomere. Myosin head will pull that thin filament towards the center of the sarcomere, and then what's going to happen? Calcium released. So once, this is the muscle contraction, sarcomere will contract, its size will shorten, right? Then calcium release channels will get closed, calcium active transport pumps will again use ATP so that they can, bring down that calcium level. That calcium it has released, muscle contraction has started, then again this calcium will again enter in the sarcoplasmic reticulum. right? And then finally what will happen, bache? Troponin tropomyosin complex slides back into the position. So this is the complete process here. This is the complete process here. Now see this. Calcium ions, they'll release, bind to tropomyosin, site will be exposed. So when ADP and PI is attached here, cross bridge will form. When new ATP will attach, there will be the breaking of cross bridge. It will result in the muscle contraction. It will result in the shortening. It will result in the shortening of that sarcomere. Look at this. Look at this. The head part here. Look at this. So they need to bind here and the cross bridge. Then we will call it as cross bridge. So there is the sliding, right? So it will pull the actin filament along its length right towards the center. Is that clear? Is that clear? Is that clear? Ye dekho. That's how it is. That's how it is. That's how it is. So see, myosin head, right? Troponin is there. Firstly, it has to remove, right? Like it will bind to the calcium, then the site will be exposed and then it will bind. Okay, then it will bind. That's how it is. This is the diagrams. Now, focus on these points. What will happen? Because see, length of I band, I'm not talking about the length of actin filament. Just it's like actin filament, giving you the example. Actin filament is of 5 centimeter. It is placed here. Let's say actin filament, it is of 5 centimeter. It is placed here. So when I'm sliding it, it will come like this. It was here, it will come like this. But it doesn't mean, if the length of the actin filament is 5 centimeter, it will stay 5 centimeter only. When myosin will pull it towards the center, it will just bring it closer, right? It will just bring it closer. It will bring it towards the center. But the length will not change, na? Length will not hamper. Just say earlier your I band was here, right? When the myosin will pull it towards the center, it will be here. So that's why we are saying that length of I band, it gets reduced. That's why we say here that length of I band, it gets reduced. That is why we say here that length of I band, it gets reduced. It gets reduced. Are you getting my point? It gets reduced, right? Right, but length of actin will remain same. If it is 5 centimeter, it will stay 5 centimeter only. It's just that earlier it was placed here. Now I am putting it here. Earlier it was placed here. Now I am putting it here. Are you getting my point? Are you getting my point? Right? So length of I band gets reduced, but length of A band remains same. It's a PYQ. So nothing will happen to the length of A band, but the length of I band, it will get reduced. But the length of I band, it will get reduced. Overall length of sarcomere will also decrease. Length of myofibril will decrease, and Henson zone, it will disappear. So what is Henson zone? H zone is that part of the dark band. It is that part of the A band where myosin is not overlapped by actin. But when there is the muscle contraction, when there is the sliding of actin over the thick filament, then of course, the Henson zone will disappear. Right? Because these thin filaments, they are going to overlap the actin. That is why. Okay, so length of actin and myosin will remain same. So these are the points. This is the muscle contraction. If any doubt is there, please feel free to ask. Right? Feel free to ask. These points are very, very, very important, bache. These points are very, very, very important. But your fourth point is length of myofibril decrease, length, see, shortening of sarcomere is there. So obviously there will be the shortening of the myofibril also. And when the actin is overlapping over the, when actin is sliding over the myosin, right, the myosin head is bringing it towards the center, pulling it towards the center. So Henson zone will disappear. Henson zone is that part of dark band where myosin is not overlapped by actin. But this is not going to happen when there is the muscle contraction. So overall length of actin and myosin filament will remain same. But when I'm saying I band, I'm talking about the position of that actin and myosin. So if it is 5 centimeter, earlier it was placed here, right? Earlier it was placed here, right? Now, because a myosin is sliding it, it will bring like this, but the dimensions will remain same, na, length will remain same, na. It's just that earlier it was here, now I am keeping it here. That's the meaning. Okay, that's the meaning, right? So when ATP is attached, the cross bridge will break. So this is all about the muscle contraction. This is the diagram here. Please again observe it. So when ATP is attached here, this cross bridge will break. When ADP and P is attached here, the cross bridge will form. Okay, the cross bridge will form. So I have added many diagrams here so that you can understand this particular topic, right? So this is all about the muscle contraction, and the points that I have shown you, that these are the points, these 6 points, just keep them in your mind. Trust me, the topic, you will never ever mark incorrect answer if you will keep it in your mind. So contraction is basically the change, right? How this contraction is taking place, it is just the change. It is just the change in the position of thin filament. Right, this contraction is due to the change in the position of thin filament. That's what you need to keep in your mind. Okay, okay, done. So do read it, bache. So that's how it is. Baki, this is all about the muscle contraction. Do observe the pictures here. So ye dekho, 2 sarcomeres, right? The muscle contraction has started. H zone is there. H zone is there. When the muscle contraction will be there, when it is fully contracted, no H zone. So myosin is overlapped by the actin. Myosin is overlapped by the actin. Observe it. That's the meaning here. That's the meaning here. Okay. That's the meaning here. Take. Chaliye. So let's move to the next part. So, so the reaction time of the fibers can vary in different muscles, right? Reaction time can vary in different muscles. Okay. Okay, so as I said, after muscle contraction, calcium ion will immediately enter in the sarcoplasmic reticulum by active transport. That's important, right? That's important. So troponin, tropomyosin, they will come back to their original position, and again, bache, the muscle will relax, the cross bridge will break, so relaxation will occur. Take, the relaxation will occur. Is that clear? Is that clear? Right? Take. So before going forward, we'll discuss few points with you all. So Baki, you know that there is something called Cory's cycle. Then I'll start that NCERT part again. What is it? It is the Cory's cycle. What is it? It is the Cory's cycle, right? Cory's cycle. So bache, it operates in both. It operates in both liver and muscles. It operates in both liver and muscles. See, I told you about what? I told you about the lactic acid formation. I told you about the muscle fatigue. Right? Now let's say muscles. We are using them a lot. Right? What is going to happen? They need their energy. They need their energy. They are getting the glucose. They have the myoglobin, which is basically the storehouse of oxygen. And we know that muscles, they also store the glycogen. Right? They are getting the glucose. Okay, fine, they are working. But now when their energy demand is more, they switch to the anaerobic mode. They switch to the anaerobic mode. Anaerobic, no oxygen is there. Right? You can say that when muscles are having less oxygen supply. So when they switch to the anaerobic mode, so you know that normally what is happening? Normally what is happening? You know about the glycolysis? Do you know about the glycolysis? The glucose. Right? After 10 steps, it will give us 2 pyruvate. Right? That is the glycolysis now. 10-step process it is. Oxygen independent process it is. Takes place in the cytoplasm. It is the first step of respiration. So these 2 pyruvates, if oxygen is unavailable, if oxygen is not there, means anaerobic conditions are there. Either it can go for alcoholic fermentation or lactic acid fermentation. So this pyruvate, it gives us the lactate. This pyruvate, it gives us the lactate. Right? Lactic acid dehydrogenase enzyme is there and it will give us the lactate. So that when there is the accumulation of that lactate, in the muscles, it results in the accumulation of lactic acid results in the muscle fatigue. It results in the muscle fatigue. Are you getting my point? It results in the muscle fatigue. Now what's going to happen here? Just say, let's say, see, muscle glycogen, right? You can write it, the muscle glycogen, right? 1 second. Right, make sure you write it, you will understand it then, right? Do not mark the arrow position yet, I'll tell you about it. Right, and that blood will pass it to the liver. So now here there will be 2 steps, right? So firstly write it down, then I'll explain it. Firstly write it, then I'll explain it. Firstly write it, then I'll explain it. Dekho, what's going to happen? Now, in the muscles, lactic acid is there, right? We cannot keep that lactic acid there in the muscles. So that lactic acid from the muscles, it will be passed to the blood. Blood will carry that lactic acid to the liver. Here in the liver, what's going to happen? 1/5 of the lactic acid, it will be oxidized. It will release carbon dioxide and water. But remaining 4/5 of lactic acid, it will be converted into the glycogen. Obviously, there are processes there. It will be converted into the glycogen. That glycogen will again provide the blood glucose. Then again that blood glucose will further go to the muscles, right? Then again from that glucose glycogen will form, and that's how right bache, that's how this Cori's cycle, it operates both in liver and muscles. Okay, right? So that's how the accumulated lactic acid will be again converted back to the glucose and then to the glycogen. Is that clear? Is that clear? Is that clear? Yes, Baki, when you talk about the muscles, obviously when you talk about the stimulus, the threshold stimulus is required. Like there is a minimum requirement, right? The minimum value, the stimulus. It should not be like any stimulus can start the muscle contraction. No, the minimum value, threshold stimulus, that minimum value. If it is that minimum value or above, then right, then the muscle will start contracting. So it is the minimal stimulus required to cause muscle contraction. What is it? It is the minimal stimulus required to cause muscle contraction. These are the terms that we use. Minimal stimulus required to cause muscle contraction. That is the threshold stimulus. Baki bache, muscle fibers, they also follow all or none principle. Means when they contract maximally. It's not like that little bit contracted, more contracted. Nahin, all or none principle, right? All or none principle. It's like black or white, no gray, right? So all or none principle mein what is happening? What is happening there in all or none principle? All or none principle mein beta, it will contract maximally. It will contract maximally. When muscles are contracted, they are contracting maximally or they are not contracted at all, right? So this principle is applicable on the muscle fibers, be it skeletal or smooth or cardiac muscle fiber. What is the term that I'm using here? I'm talking about the muscle fiber, the anatomical unit. I'm not saying muscles. I'm not saying muscles. I am not saying muscles here. I am saying that all or none principle is applicable on skeletal, smooth, cardiac muscle fibers. When they contract maximally. When they contract, right bache, it contract maximally. Okay. Okay, so it is not applicable to muscle and nerve, but it is applicable to muscle fibers. It is applicable to muscle fibers. Understood? It is applicable to muscle fiber. So, and it is not like that if one stimulus has passed, muscle is contracting, then next will immediately pass. No, you know that it takes time to recover as well, no? Chaliye. So here you can see the reaction time, it vary in different muscles. When there is the repeated activation of muscle, it can result in the accumulation of lactic acid. I told you about it. I told you about the Corey cycle. I told you it will operate in muscle and liver. Do remember that. So when there is the anaerobic, means in the absence of oxygen, when there is the breakdown of glycogen in muscles, it will result in fatigue, right? Now, here they are talking about the red muscle fiber and white. So muscles contain red colored oxygen storing pigment. Remember, hemoglobin, it transports the oxygen, right? It transports the oxygen. Myoglobin, it stores oxygen. It stores oxygen, right? Myoglobin is a tertiary protein. It stores oxygen. It is an important point. It's the oxygen storing pigment. It is the oxygen storing pigment, right? Oxygen storing pigment. Now myoglobin content, if it is high, then muscles will be reddish in appearance. That muscles are red fibers, right? So these muscles which are having high myoglobin, they are red muscle fibers and they have plenty of mitochondria. More mitochondria means more ATP, but they have less sarcoplasmic reticulum, right? So they have plenty of mitochondria, so they use large amount of oxygen stored in them for ATP production, right? So therefore they are the aerobic muscles. So red muscle fibers, aerobic muscle fibers having high myoglobin content, high myoglobin, high mitochondria, but they have less sarcoplasmic reticulum, right? They have less sarcoplasmic reticulum. So the muscle fibers which are having less quantity of myoglobin, they appear pale or whitish, they are white fibers, number of mitochondria are few in them, but the amount of sarcoplasmic reticulum is high. So means contraction will occur immediately here, right? So these are they depend upon anaerobic process. Okay, okay. Yes, okay. So in red muscle fiber, slow and sustained contractions will be there. Slow and sustained contractions will be there in slow and sustained contractions will be there. And of course, because in the white muscle fibers more sarcoplasmic reticulum is there, so contractions will be fast, they will be for the short duration. So that muscles, they get fatigued easily. Okay, okay. So there are examples, जैसे extensor muscles of back, they are the example, and there are many other examples as well. Fine. Chale. So next part is the skeletal system. So say hi to Chintu. So do you need break or we can just finish it? Do you need break or we can just finish it? Hello, hello. Tell me quickly, do you need break or we can just finish it in one go? Dinner break. Chalo. So let's meet at 8:45. Done? See, we will go as per majority, okay? Hello, am I audible? Yes, am I audible? No need to make a missing poster. Chalo. Now, okay, chali, let's continue. So now we will talk about the human skeletal system, and as I said, it is not difficult. Trust me, it is not difficult. Okay, so what are we going to talk about? We will talk about the axial skeleton, appendicular skeleton. There are total 206 bones when we talk about the human adult. How many bones are there? How many bones are there? Yes. Now we have to act like a team, right? I'll ask the questions, you have to answer it. Okay, you have to answer it. Okay, right? So in the human adult, how many bones are there? How many bones are there, bache? 206 bones are there. How many bones are there? 206 bones are there. Is that clear? Sure? Sure? Chale, let's continue. Okay, but bache, so here 206 bones are there in the human adult, but in the embryo the number of bones are different. They are around 200, they are around 300, right? But in adult 206 bones are there. So look at this skeletal part, right? Look at this. See, when you talk about this axis, right? This is the axial skeleton. Axial skeleton. You take the example of a doll, right? Imagine a Barbie doll. So from that Barbie doll, if you remove the arms and the legs, the remaining structure that is left will be the axial skeleton. Of course, when you are talking about us, when you are talking about humans, here again we have to remove the forearms, right? We have to remove the girdles, we have to remove the hind limbs and the girdle, and the remaining portion will be the axial skeleton, and these limbs and girdles, they will make the appendicular skeleton. So look at it, this is what we need to remember. So how many bones are there? 206 bones are there. 206 bones are there. So we will discuss the number also. So skeletal system consists of a framework of bones and a few cartilages, right, bache? So what do we have in our skeletal system? Our skeletal system is also mesodermal in origin. It is also mesodermal in origin. So it contains bones and it is having the few cartilages as well. It contains bones and it is also having the few cartilages as well. Yes, bache? Yes, bache? Understood? 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 So imagine chewing food without jaw bones and walking around without limb bones. Of course not possible. So bones and cartilage, what are they? They are the specialized connective tissue, right? They are the examples of connective tissue. They are the specialized connective tissue. So that is your homework. You will write down the difference, right, bache? You have to write down the difference in bone and the cartilage. Okay, this is your homework. What is the difference in bone and the cartilage? That is what you have to write. So it is the specialized connective tissue, my dear students. Bones are having the hard matrix, right? They have the calcium salts. They have the hard matrix, right, bache? And this cartilage, it is flexible. It is pliable. So this cartilage, why is it pliable? It is having the chondroitin salts. Chondroitin salts are present there in the cartilage. So C for cartilage, C for chondroitin salt. And what do we have in the bones? Bones are having the calcium salt, right? There are other differences also that you need to read. Just say the cells that make the bones. We study that in structural organization in animals. The macrophages of that or you can say that, right, the bone destroying cells, cartilage destroying cells, all that points are important. Do read about it from the chapter structural organization in animal. I'll stick to this chapter here only, right? So as I said, adults will have 206 bones, embryo will have around 300 bones, right, bache? So it is grouped into two divisions. So here let's discuss that. We have the axial skeleton, we have the appendicular skeleton. What do we have? We have the axial skeleton, we have the appendicular skeleton. Axial skeleton having 80 bones, appendicular skeleton having 120 bones. Again, I'm repeating it. See, if you will hate this topic, yaar, madam, human skeletal system, I cannot remember, so many bones are there, then trust me, you will never ever be able to understand this topic. But if you have that zeal, nahi yaar, I will remember it. And it's interesting as well. Actually, I don't have the skeletal system here, otherwise I would have shown you this, but still, I will try my best to make it very easy for you. So again, I'll suggest you one thing. You guys can watch my Game of NEET lecture if you want a detailed, explanation of this part. This is a one shot and it is a very quick wala one shot. Okay, we will deliver all the content, but if you want very slow explanation, you can watch that lecture. So I'll try my best to make it easy for you. So please pay attention, please focus here. Axial, appendicular, right? So axial skeleton will have skull, it will have skull. Right? What will it contain? It will contain skull first. Or in the skull, what will be discussed? We'd say 29 bones are there in totality. Now you will be like, yeah, NCERT is saying 22 bones. I'll clarify everything. So skull you will talk about the cranium, and we will also talk about the facial bones. Other than that, here in axial skeleton, overall I'm telling you about the axial skeleton. So what do we see in the skull here? What are we going to see in the skull here? So in the case of skull, we will talk about the cranium, we will talk about the facial bones, and here itself we will talk about the ear ossicles, right? Ear ossicles. And you know that in the books, the hyoid bone is also mentioned here in the skull only, right? Hyoid bone is also mentioned there as the part of skull only. But here I will it is the hyoid bone is the part of axial skeleton only, but here I'll provide you one correction, right? Here I'll tell you about one correction, hyoid bone. So when you talk about this hyoid bone, bache, when you talk about this hyoid bone, it is a small U-shaped bone which is situated in the midline of, midline of neck. Where is it situated, bache? It is situated in the midline of neck. You can write down this point right now. Let me explain the things slowly, then you will understand everything. So it is situated in the midline. It is situated in the midline of neck, and it is anterior at the base of mandible and posterior at fourth. It is anteriorly at base of mandible. It is the lower jaw, right? And posteriorly at fourth cervical vertebrae, right? There is a correction regarding that. We will discuss it, but right now this is what you can write down, and then I'll explain it, right? So anteriorly at the base of mandible, posteriorly at fourth cervical vertebrae. Take fourth cervical vertebrae. So first of all, focus on the numbers here. As I said, skeleton 206 bones, axial 80, appendicular 120, axial 80, appendicular 120, axial 80, appendicular 120. We are talking about the skull here. So when we are talking about the skull, I told you cranium, facial bones, ear ossicles are there, right? Ear ossicles are there. So when you talk about the cranium, right, how many bones are there in cranium? Yes, everybody. How many bones are there in cranium? In the cranium, we have the 8 bones. Facial bones are total 14, and ear ossicles are total like 2 into 3. Means each ear, middle ear, is having 3 ear ossicles. 3 ear ossicles. Individually also I will discuss. So hyoid bone is also 1 in number. Hyoid bone also 1 in number. So in totality, we say 29 bones are there. Right, so this is about the axial skeleton we are discussing. We are talking about the skull. So when you are saying skull, cranium, facial bones, ear ossicles. Cranium, facial bones, ear ossicles. Cranium, facial bones, ear ossicles. And hyoid bone, I told you about its position. Right, then other than that, in the axial skeleton, what else do we discuss? What else do we discuss here? I hope you have mentioned everything related to the hyoid bone, that it is situated in the midline of neck, anteriorly at the base of. So it is present here at the base of mandible, posteriorly at fourth cervical vertebrae. Right? I hope it is done and dusted. Now, my dear students, in the axial skeleton, what else do we discuss? We talk about the vertebral column. Right, we talk about the vertebral column. So total 26 vertebrae are there. So it is CTLSC, 7, 12, 5, 5 and 4, right bache, right? So vertebral column 26. Then here we also talk about the sternum and we also talk about the ribs, right? So sternum 1, ribs total 24. This is what we discuss in the axial skeleton. Again as I said here, what are we supposed to do? We just need to revise it again and again. We just need to revise it again and again. If the number of bones is clear to you now, everything will be easy for you. Axial skeleton, 80 bones. How many bones are there? 80. When you are talking about the skull, what you need to discuss here? You will talk about the cranium. 8 bones. Facial bones, how many bones are they? They are 14, right? They are 14. When you talk about the ear ossicles, right? When you talk about the ear ossicles, how many ear ossicles are there? 3 into 2, total 6, right? Miss has malus, incus and stapes, right? We'll come to this. So this is what we discuss here. And hyoid bone may, as I said in the NCERT, it is considered as a part of skull. It is present at the base. So like that's why I'm writing it separately so that there should be no confusion. Other than that, in the axial skeleton, as I said, what do we discuss? We talk about the vertebral column. How many vertebrae do we have? 26, right? Then sternum, which is also known as breastbone, you know its role in the it will form the rib cage, how well it protect, you know it. So then comes the ribs. So when you talk about the ribs, there are total 12 pairs. So 12 pairs means total 24 ribs are there. That's how you have to remember. This is about the axial skeleton. Now let's talk about the appendicular skeleton. Let's talk about what? Let's talk about the appendicular skeleton. So students, when we talk about the appendicular skeleton, what will you discuss? As I said, we will talk about the girdles here and we will talk about the limbs. So girdles mean shoulder girdle, that is pectoral girdle, right? There will be the shoulder girdle, that is pectoral girdle, and there will be pelvic girdle, that is the hip girdle, So pectoral girdle is having total 4 bones, right? 2 clavicle, 2 scapula, and here you talk about the 2 bones, and that are coxal bones, also known as innominates. So if I can remember it, you can remember it too, and trust me, it's easy. So there are 2 coxal bones and they are formed by the fusion of each coxal bone So each coxal bone is formed by fusion of ilium, ischium, pubis. Ilium, ischium, pubis. Now you tell me, here it should be I-L-E-U-M or I-L-I-U-M. Here it should be I-L-E-U-M or I-L-I-U-M. Ilium. What should be the spelling of ilium here? I-L-E-U-M or I-L-I-U-M? Bache, it should be I-L-I-U-M because I-L-E-U-M is there in the small intestine, right? It is there in the small intestine. Is that clear? Is that clear? So ilium, ischium, pubis. So it is I-L-I-U-M. It is not I-L-E-U-M. I-L-E-U-M is the part of small intestine. So pectoral girdle, right? 4 bones, 2 clavicle, 2 scapula. Pelvic girdle, 2 bones. That is coxal bone or innominates. They are formed by the fusion of ilium, ischium and pubis. And then we talk about the limbs here. What do we discuss here? Limbs. So 6 bones here. Total in appendicular skeleton 126 bones. So limbs will have 120 bones. Limbs will have 120 bones. So 4 limb maybe. I'll discuss the division here also. Then you have the hind limb also. 4 limb and hind limb. 4 limb is also having 60 bones. Right, hind limb is also having 60 bones. One by one we will discuss it. That's what we have to study. That's what we have to study. Now come to the cranium part directly. Come to the cranium part directly. At least remember the number of bones. At least remember the important bones. That is my advice. Right? At least remember the important bones. At least remember the important things, right? Important points regarding this also. So cranium, how many bones are there? 8 bones. So cranium is the brain box, bache, right? It is the brain box, bache. It will protect our delicate brain. It will protect our delicate brain. So frontal, parietal, temporal, occipital, ethmoid, sphenoid. Frontal, parietal, temporal, occipital, ethmoid, sphenoid, right? So when you look at the skull, when you see, this is the outer part, these are the bones here. You will see that skull bones, they will not show any movement. They are just attached to each other. So here, right, joints are present. Which joints are they? They are the immovable joints. They are the immovable joints. We will discuss the joints here in this chapter also. So they are the immovable joints, and we use the word sutures for it. What is the word that we use for it? We use the word sutures for it. So what are the name of the bones is frontal, parietal, temporal, occipital, ethmoid, and sphenoid. So 1, 2, 1, 1 and 1. Total 8 bones, right? Total 8 bones. So look at this. This one here is occipital, right? Temporal, near to the ear here, right? This one is the parietal. So frontal, like see, this is this part, frontal, parietal, this temporal, right? Occipital, then ethmoid and sphenoid. Will show you the position as well. Ethmoid and sphenoid will show you the position as well. So when you talk about the sphenoid bone, S for sphenoid, this is the detail that you should know. S for sella turcica. S for sphenoid, S for sella turcica. Actually, here in this bone, there is a kind of depression. There is a kind of cavity. There is a kind of depression. There is a kind of space there. And that is sella turcica, and it is the house of pituitary gland. So pituitary gland, the master gland, the master endocrine gland will be there in this sella turcica. Sphenoid bone it is, right? Sphenoid bone it is. Now, the second thing is occipital. Now in the occipital bone, when you look at this part, just say you know that this is a skull and this is the bony skeleton. The skull is protecting our brain, right? And we have the vertebral column that will protect the spinal cord. So skull, in this particular part, we will use the word articulation, right? We will use the word articulation repeatedly. We will use the word articulation repeatedly. Articulate, that bones will articulate. They will join with each other. Then only the movement is possible. Just let's say, I am moving my neck like this. Why? Because my skull, it is articulated with the vertebral column. The first vertebra is atlas, the second is axis. So skull, it articulates with the atlas, right? Then second vertebra is axis. So articulation is the word that I will use. Now you know that when you talk about the brain part, the medulla oblongata, it will further continue in that spinal cord. We know the spinal cord also. So now in the occipital bone, you will see a kind of opening, a kind of aperture there. We call it as foramen. Yes, what do we call it? Foramen magnum. What do we call it? Foramen magnum. Yes or no? This opening, what do we call it? Yes, people, what do we call it? We call it as foramen magnum. Am I right? Do not confuse it with the foramen ovale or foramen monero. So it is the foramen magnum. Right? So here to its sides, you will see condyles. There are 2 condyles. So these condyles, they are in the occipital bone. They will articulate with the first vertebra, right? So first vertebra is atlas. Number 1. So from this foramen magnum, that spinal cord will come out. That's what you have to remember. So this is the detail, at least that this detail should be clear to you. Okay. So cranium, 8 bones, parietal 2, frontal 1, ethmoid. Here ethmoid is present, occipital, temporal, sphenoid. And as I said, when it comes to the facial bones, they are total 14. How many bones are there? 14. Just these cheekbones, zygomatics. You also talk about the jaws, 2 maxillae, 1 mandible, right, bache? Other than that, we also discuss about the palatine. We also discuss about the lacrimal. There's a tear gland near to this part. Lacrimal is there. Nasal, palatine, zygomatic, inferior nasal concave, vomer, maxilla and mandible. Right? Vomer, maxilla and mandible. Now, these are the facial bones. So these cheekbones are what? Zygomatics. Right? Nasal. From nose, nasal word. From the tear gland, the word lacrimal should be there in your mind. The palatine is also there. Right, bache? So, upper jaw is made by the fusion of 2 bones. Maxilla. Upper jaw is maxilla made by 2 bones. 2 bones will form our upper jaw. And mandible And when it comes to the mandible, so mandible, that is lower jaw, so mandible is 1. So posterior to this, so you know that at the base of it, the hyoid bone is present. So one more thing that you need to remember, that mandible is the only movable bone of skull. It is the only movable bone of skull. Mandible is only movable bone of skull. It is the only movable bone of skull, right? So look at this nasal here in the nose, lacrimal somewhere here, right? Inferior nasal concave, they are also 2 in number. Palatine here, zygomatic, that cheekbone, and this yellowish part is the maxilla, 2 in number. Mandible is 1, vomer is 1. So here I just remember one thing, that when we talk about the facial bones, only mandible and vomer, they are single. Rest all are double, double, double. Rest all are double, double, double. You read these words, right, twice or thrice. Trust me, they will be on your fingertips. Trust me, they will be on your fingertips. Baki, we know that when it is the mandible and maxillae, they are also having the spaces. We call them, right? We call it as alveoli, and the teeth will be embedded there. Okay? Okay? Yes. So these spaces here, or you can say these sockets here, where we call them as alveoli. We call them as alveoli. Understood? We call them as alveoli. Understood? So sphenoid clear. Sella turcica clear, these facial bones clear, right? Sphenoid clear, sella turcica. So I have mentioned all the important points. So sphenoid, sella turcica, and yes, it's a butterfly shaped bone. It's a butterfly shaped bone. What kind of bone it is? It's a butterfly shaped bone. So skull mein we are talking about the cranium, 8 bones, facial bones are 14 in number, right bache? Now let's talk about and mandible is the only movable bone of skull. So let's have a look at this occipital bone part here. So in this occipital part, this opening, this wide thing here, right? This wide thing here, this is the foramen magnum. This is the foramen magnum through which that medulla spinal cord will come out. And here, this part is the occipital condyles. So our skull is dicondylic. 2 condyles are there. So we have dicondylic skull, right? In some animals, it is monocondylic as well, but we have dicondylic skull. 2 condyles are there. They will articulate with first vertebra atlas. They will articulate with first vertebra atlas. Ye dekho, right? Watch it. Posteriorly, how the things are there, foramen magnum. And what do you have there? Which joint do you have here? Which joint do you have here? Which joint do you have here? Yes, please, immovable joint. We call it as suture. Immovable joint it is. We call it as suture. Right, bache? Right, bache? So I'm writing it here. This joint is immovable. Right? Immovable joint. You can also call it as fibrous joint, and there is another word for this, that is synarthrosis, right? It is the fixed joint as well, and there is one more word, synarthrosis, right? Synarthrosis, that is also the word for this joint here. Is that clear? That is also the word for this joint here. Is that clear? Is that clear? Matlab, let's discuss it here only now. Why to discuss this topic separately? Okay, so it is the synarthrosis joint. It is the synarthrosis joint, right? Right. So it will not allow the arthrosis. It is na. It doesn't allow movement. It doesn't allow movement. Right? It doesn't allow movement. Understood? Understood? So the skull bones, they are fused end to end with the help of these, right? They are fused end to end. How? With the help of these dense fibrous connective tissue. So they are immobile. This is the joint here. So joint part clear, foramen magnum clear, occipital condyle clear, sutures clear. Yes, sutures clear. Yes. Sure. Any doubt? Any doubt? Just read these bones again and again, right? Remember their names, right? Right, remember their names. So skull is dicondylic, and dicondylic skull, जैसे you will see skull is dicondylic in mammals and even in amphibians. In amphibians also it is dicondylic, and in birds and reptile bache it is monocondylic. So frog amphibian is the part of your syllabus. So yes, you should remember this. So repeat this. Trust me, they are not difficult. You will get used to it. You will get used to it. So this is the first vertebrae, atlas. So here that occipital condyles will join. Are you getting my point? So look at this vertebra here. The space here is vertebral foramen, right? The space here present in is vertebral foramen, this space, right? Neural canal is also the word that we use. These are the transverse processes. So just look at it. Any doubt? Any doubt? Any doubt? Skull part clear? Chalo. So after skull now, and yes, how can I forget about the ear ossicles? How can I forget about the ear ossicles here? I didn't mention the ear ossicles here. So let me mention this. So ear ossicles. So bache, see, in each ear, in our middle ear, these bones are there. So the trick here is Miss has. What's the trick? The trick here is Miss has. It is malus, it is incus, it is stapes, right? So Miss has is the trick. Miss has is the trick. So H stands for hammer shaped, A stands for anvil shaped, it is stirrup. So yes, that's important. Ear topic is deleted from your syllabus, but ear ossicles are not. So these are 3 ear ossicles, and the stape, it is the smallest, ear ossicle. It is the smallest ear ossicle, plus it is the smallest bone also. So if in the paper the question is smallest bone of our body, that's stapes. So they are present in the middle ear, they will amplify the sound. So Miss has, malus, incus, stapes. Miss has, malus is hammer shaped, incus is anvil, stapes is stirrup. Malus is hammer shaped, incus is anvil, stapes is stirrup. That's what you need to remember. Ear ossicles be done. Now before going forward, I would like to tell you about the hyoid bone. So as I said, right, as I said, it is U-shaped bone. What kind of bone it is? It's a U-shaped bone, right? It is the, right, look at the structure here. This part is important. Kindly pay attention, right? Kindly pay attention. This part is important. You are talking about the hyoid bone, and there is a special thing about it. This is the bone which will not articulate with any other bone. Do you know that? It is the bone which will not articulate with any other bone. It will not articulate with any other bone. Wait, let me explain this to you. So till then you please write it. Okay, so what are we talking, guys? What are we talking? We are talking about the hyoid bone, which is also the U-shaped bone, right, bache? So it is known as the tongue bone. It is known as tongue bone. And remember, your tongue, why? Because the tongue is attached. Tongue is attached to hyoid bone. Tongue is attached to hyoid bone. And let me tell you, this bone, it is the only bone that doesn't articulate with. It doesn't articulate with any other bone. Do you know that? It doesn't articulate with any other bone. It is not attached with any other bone. See, hyoid bone, then this membrane, then the larynx, the thyroid cartilage, the largest larynx, the largest cartilage of the larynx. That's how it is. So tongue is attached to it. So I told you about its position. Just say it is written, it is present at the base of the mandible, right? It is written, bache, that it is present at the base of buccal cavity, and it is also included in skull. But as I said, correction is there. So always remember, it is situated in the midline of the neck. It is situated in the midline of the neck, anteriorly at the base of mandible, posteriorly at 4th cervical vertebrae. Posteriorly at 4th cervical vertebrae. Understood? Understood? Right? Ear ossicles clear. This diagram clear. So frontal, this frontal, right? Then green part is the parietal, right, bache? The temporal will be the red part, and then there is the sphenoid and ethmoid. Then there is the sphenoid and ethmoid, right? So that's how the skull articulates with the vertebral column. Ye dekho, this is the first vertebra. What do we call it? We call it as atlas. So now we will talk about the vertebral column. Now we will talk about the vertebral column. So for the vertebral column, you have to remember one thing, that is come to lunch, stay calm. Come to lunch, stay calm. Come to lunch, stay calm, right? So this is the vertebral formula. So smallest vertebrae, your cervical vertebrae, then cervical, thoracic, come to lunch, lumbar, sacrum, ya sacral, and then comes the coccyx, ya coccygeal. So it's 7, 12, 5, 5, 4. That's how you have to remember. 7, 12, 5, 5, 4. 7, 12, 5, 5, 4. 7, 12, 5, 5, 4, right? It's 7, 12, 5, 5, 4. So if you will notice here, when I'm saying 5, 5, 4, the bracket is there. This bracket is showing the fusion. So it means after fusion, matlab before fusion 5 bones were there, after fusion they will form 1. So that is how we write it. So that's what you can say, that how many bones are there in the vertebral column of embryo. So before fusion it will be like this: 7, 12, 5, 5 and 4, right? So total it will be, yes, total it will be, yes, 7, 12, 5, 5, 4, total it will be 33. Am I right? Before fusion it is 33. Is it 33? Are you add it now? 33. And after fusion what will happen? After fusion what will happen? It is again CTLSC. So you can write it like this: 7, 12, 5, 5 and 4. But these 5 they will fuse to form 1. These 4 they will fuse to form 1. Right? So total how many will you have? 26. Okay. Okay. 26. Is that clear? It will be 26. Is that clear? Is that clear? So just look at this part. Just look at this part. Cervical vertebrae 1 to 7. So mostly in the mammals this is what you will see, and they are the smallest vertebrae. So first here is atlas, second here is axis. First here is atlas, second here is axis. Is that clear? Is that clear? So in almost in all the mammals, you will see that 7 vertebrae are present. Take 7 vertebrae are present. Is that clear? Okay. Okay. Oh, sorry. First is atlas, that will articulate with occipital condyle, and second is the axis here, right? So atlas is having an opening bache, odontoid fossa, in which the projection of axis will fit and they form the pivot joint. ठीक? जैसे atlas is the first and second is axis. So as I said, here it is having one odontoid process where, you know, axis will fix. So basically they form the pivot joint. They form what kind of joint? Pivot. Why am I discussing the joints here? So that, you know, joint is also the part of your syllabus. So if you will write it here only, you will understand that in a better way. ठीक है? ठीक है बच्चे? Okay. So there is odontoid fossa in the atlas, right? Basically, yes. Atlas is having the opening odontoid fossa in which projection of axis will fit, right? So which joint are they going to form? They will form the pivot joint, and that pivot joint, a pivot basically, pivot you will say, pivot you will say, not the pivot, pivot you will say, pivot joint it is going to form, and it is the example of movable joint. It is the example of movable joint. I'll discuss the joints as well, but you write it. You write it here, right? So it is the pivot joint. It is the pivot joint, okay? What is it? It is pivot joint, movable joint, synovial joint, right? So pivot joint is the example, right? It is the example of the movable joint, and movable joints are also known as synovial joint. So it is the pivot joint, the joint in between atlas and axis. So do remember the examples here. Do remember the examples here. So firstly, tell me this part clear. Then thoracic vertebrae. You know that these thoracic vertebrae, they will be there in the rib cage. We'll come to that part. The lumbar, and then there is the sacrum. 5 will fuse to form 1. 4 will fuse to form 1. Okay? Now look at this part. This is the right lateral view. This is the right lateral view. And this is the posterior view. First of all, look at it. Then we will discuss. Right? First of all, look at it. Then we will discuss. Do it quickly. Do it quickly. Okay. Right. So when you look at these vertebrae, right, when you look at these vertebrae, just say this is one, this is second. So body of vertebrae, for that we use the word centrum. For body of vertebrae, what is the word that we use? Centrum. Just say this is the complete body of the vertebrae, this is another. So in between these vertebrae also, na, in between these vertebrae, that is also PYQ, bache, you will see something that is known as yes, bache, what is it? That's a homework. Sorry, that's an important question. What is it? here in between these two, what will you see? You are going to see something that will connect the centrum. That will connect the centrum. So what is that, bache? It is the inter means in between, intervertebral disc, and that is nothing but a cartilaginous joint. Right, cartilaginous joint. It's a type of joint which will provide a little movement, right? So it is intervertebral disc, which is the example of cartilaginous joint. It is the intervertebral disc which is the example of cartilaginous joint, and it is, or you can also write, it is the fibrocartilaginous joint. What is it? It is the fibrocartilaginous joint, and let me tell you, it is only in mammals. It is also unique in mammals. You will see it only in mammals. It acts like a shock absorber. It acts like a shock absorber. They all are previous year questions, right? They all are previous year question. So these are the, this is the fibrocartilaginous disc, yeah, fibrocartilaginous joint. Chalo, disc is the appropriate word here. You can write it. Take, so it acts like a shock absorber. It is only there in the mammal. So centrum is the body of the vertebrae. So the centrum, right? It is, this disc is present between centrum of vertebrae. That's what you need to write. Is that clear? That's what you need to write. Is that clear? Is that clear? Sure. Any doubt? Any doubt? So vertebral column will protect spinal cord, it support our head, it serve as the point of attachment for ribs and muscles, right? Right? Yes. So if you will see cervical and lumbar curve, right? Now see, our vertebral column is curved. Our vertebral column is curved, right? This is actually, so cervical and lumbar. This curve is directed forward. Please pay attention. Cervical and lumbar curve, they are directed forward. They are, this information is important, they are directed forward, right? And basically your this thoracic curve and sacral curve, it is directed backward. It is directed backward. So one is directed forward, another is directed backward. Take, and this is very important. Why? Because this lumbar curve, you know that when we talk about the evolution, right, initially even, right, we were not having the bipedal locomotion, right? We were having the quadrupedal locomotion. When you talk about this evolution part, we evolved from the organisms having the quadrupedal locomotion. So when it comes to bipedal locomotion means we are using our four limbs, not like dogs that we are using all our limbs for walking. We are using our four limbs for the walking, right, bache? So this lumbar curve, right, this lumbar curve, the curve in this lumbar, it helps in bipedal locomotion. It helps in bipedal locomotion. Understood? It helps in bipedal locomotion, right? And because of this lumbar curve, bache, the posture is also, right? Erect posture we have. What kind of posture do we have here? Erect posture we have, right? Erect posture we have here. Okay, so look at this. Look at this. This is the vertebral foramen, also known as neural canal. Overall body is the centrum, right? And as I said, in between, what will you see? You will see intervertebral discs. That is only in mammals. That is only in mammals. Any doubt here? Any doubt here? Any doubt here? Sure. So vertebral column done. Sternum. Baki sternum, you know that it is the flat bone on the ventral midline of the thorax. Next is the sternum. Next is what, bache? Sternum. It is kind of dagger shaped, right? It is the breastbone. It is dagger shaped flat bone on the ventral midline of thorax. Look at this. This part. When you talk about it, we say it is dagger shaped. This kind of shape is dagger. So it is dagger shaped, present in the midline of the thorax, bache. It's a flat bone. It is the breastbone, right? It is the flat bone. It is the breastbone. Look at this part. So this tie-like part, this is look like the note of the tie, na, manubrium, right? This is the body. This is the body here, and this one is the xiphoid process. This is the sternum. Flat bone. It is dagger shaped. It is dagger shaped, right? Flat bone it is. Okay, so basically in the case of sternum, we know it very well, right, that it takes part in the formation of rib cage, okay, that thoracic basket and rib cage, it plays role in respiratory mechanism as well. Now we will talk about these ribs. Look at the structure here. Now what we have to discuss, we have to talk about the ribs here. So as I said, how many pairs do we have? 12 pairs. 12 pairs means total 24 ribs are there, right? So guys, I will relate it with the breathing and exchange of gases chapter also. So you know that when we talk about the rib cage, so rib cage is also known as thoracic basket. Rib cage is also known as thoracic basket, right? So when you talk about it, you know that ventral side will have, जैसे dorsal side will have vertebral column, specifically thoracic vertebrae. Ventral side will have sternum. Ventral side will have what? Sternum. And laterally you will see ribs, right? Laterally you will see ribs. Yes, laterally what will you see? You will see ribs. So that's what you need to remember. And if in the paper they are asking about, see, vertebral column, maybe if we have to be specific, it is the thoracic vertebrae. What's that? It is the thoracic vertebrae. Now let's talk about the ribs. When you talk about the ribs, as I said, how many pairs are there? 12 pairs. How many pairs are there? 12 pairs. So first to seventh, they are the true ribs. 8th, 9th or 10th pair, they are the false ribs. This is important. 11th and 12th pair, these are the floating ribs. They protect the kidney actually. They are the floating ribs. Actually, bache, when you talk about the ribs, dorsally they are attached to the vertebral column. Ventrally they are attached to the sternum. Dorsally they are attached to the vertebral column. Ventrally they are attached to the sternum. So first to seventh pair of ribs, it is attached dorsally to the vertebral column, ventrally to the sternum, obviously with the help of cartilage, of course. So this is also known as vertebrosternal ribs, right? They are the true ribs or vertebrosternal ribs there, right? We call them as vertebrosternal ribs or the true ribs, right? Vertebrosternal ribs or the true ribs, right? So dorsally they are attached to the thoracic vertebrae, ventrally to the sternum, but as I said, with the help of cartilage, right? Right, with the help of cartilage. And which cartilage is there? Hyaline cartilage or costal cartilage. Matlab these first seven pairs, dorsally to vertebral column, ventrally to the sternum. So when they are attached to the sternum, they are attached with the help of hyaline or the costal cartilage. One and the same thing. Matlab cartilage again is helping there. Now 8th, 9th and 10th. Dekho, dorsally to it is attached to the vertebral column. Dorsally it is attached to the vertebral column without any doubt. But ventrally, dorsally it is attached to the, ventrally it is indirectly attached to the sternum. It is actually attached to the seventh pair and, right, it is actually attached to the seventh pair. So indirectly it is attached to the sternum, not directly, right, not directly. So ye dekho. So 8th, 9th and 10th pair of ribs, it does not articulate directly with the sternum but join the 7th rib with the help of hyaline or costal cartilage, right, with the help of hyaline and costal cartilage. So what is the word that we use for it? We call them as vertebrochondral, right? False ribs are also known as vertebrochondral ribs. Vertebrochondral ribs, understood? Vertebrochondral ribs. So they do not articulate directly with the sternum but attached to the 7th. Then the floating ribs, they are known as vertebral ribs because bache, they are not at all attached to the ventral side. They are just attached to the this part, vertebral column. So last two pairs, they are not connected ventrally, therefore they are the floating ribs and we can also call them as vertebral ribs. That's the point. So this part is very, very, very important. So if you are not capable to remember all the bones here, at least do not miss the ribs, right? At least remember ribs. Okay, do not forget about the ribs. So look at this part here. Here dekho, the first pair, it is attached to the manubrium of the sternum, right? है ना? Then this second, third, fourth. Okay, so now here the structure that you will see, this bone here, it is clavicle. It is also known as collarbone. It is the part of pectoral girdle. It is also articulating here with the sternum. That's what you can see, right? That's what you can see. So we will come to this diagram later. So I hope this part is clear. Now let's talk about the appendicular skeleton, right? This is all about the axial skeleton. Now let's talk about the appendicular skeleton. Okay, let's talk about the appendicular skeleton. So how many bones are there in appendicular skeleton? 126. Let's start with the forelimb first. So forelimbs में in totality 60 bones are there. How many bones बचे? 60. It is 2 into 30 because we have two forelimbs. We have two forelimbs. So here you will talk about humerus, radius ulna या radio ulna. Then ये देखो, this is humerus, this part here, this longest bone of forelimb I'm saying. Overall that longest and strongest bone in our body is femur and smallest bone is stapes. But I'm talking about the forelimb. I'm talking about the forelimb. It is the humerus. This part is humerus. Radius ulna, radius ulna, radius ulna, wrist bones, right? We call them as, yes, what do we call them? Carpals या metacarpals? जैसे we have carpals, metacarpals and phalanges. We have carpals, metacarpals and phalanges. So the bones of wrist, they are the carpals, right? They are the carpals. So wrist bones, they are carpals. Then metacarpals are there. Palm bones, right? Palm bones, they are metacarpals. And bache, these bones here, they are phalanges. These bones here, they are phalanges. What are they? They are phalanges. Dekho, one humerus, but we have two forelimbs. That's why multiply by 2. One radius, one ulna. One radius, one ulna. They are also having a joint. We'll discuss it. So that's why we said 2 into 2. Because there is one radius. Sorry. Ye dekho, this finger, little finger side, this is you are having ulna. Towards the thumb side, you are having radius. So, ulna, you, in Hindi, right? We call finger as ungli, right? Right, ungli. So, ulna, towards this side, towards thumb side, it is the radius. So, radius and ulna is there. Wrist bones, they are total 8 in number. So, here it will be 2, here it will be 4, 8 into 2. Total will be 16 here. Palm bones, 1, 2, 3, 4, 5. So 5 into 2 it will be, then it will be 10. When you talk about the phalanges, bache, 2, this is the phalangeal formula. 2, 3, 3, 3, 3. 2, 3, 3, 3, 3. 2, 3, 3, 3, 3. 2, 3, 3, 3, 3. Is that clear? This is the phalangeal formula. 2, 3, 3, 3, 3. Right, so matlab 3 plus 3, 6 plus 3, 9. 2, 3 plus 3, 3, 3, 3. So 3 plus 3 plus 3 plus 3. 12. What is it? Anji, what is it? We have 2, 3, 3, 3, 3. Count it. 3 plus 3, 6. It's not 9. It's 3 into 3 is 9. So 3 plus 3, 6 plus 3, 9 plus 3, 12 plus 2, 14. So 14 into 2, it will be total 28. Right? Total 28. This is about the forelimbs. Right? So 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, then 16, 17, 18, 19, 20, 21, 22, 23, 24, 25, 26, 27, 28, 29 and 30. So this is what we have. This is what we have. Is that clear? Is that clear? So 8 carpal bones, palm bones are 5 in number, phalanges are total 14. Phalanges are total 14. Is that clear? Now, let's talk about the hind limb. Let's talk about the hind limb also. So in totality, it will be 30 into 2, total 60 bones will be there. And here you talk about the femur, the strongest bone of our body. Femur, then tibia, fibula. But here, there is one sesamoid bone, that is patella. That is patella. Then, oh, let me write it properly then. Femur, tibia, fibula, patella. Then here you have tarsals. Do not confuse carpals and tarsals. right bache? These are the tarsals, metatarsals and phalanges. Tarsals, metatarsals and phalanges. And bache, for patella I have used the word sesamoid bone. What is the meaning of sesamoid bone? You should know this. It is a PYQ. Okay, so femur 1 into 2, 2. Again, tibia is also 1, fibula is also 1. Total 4. There is 1 kneecap. So total 2 kneecaps. This is patella. This is also known as kneecap. Right? Tarsals. Here tarsals, they are 7. So 7 into 2, 14. So see, humerus 1. Radius ulna 1 and 1 plus 1 plus 1 2, carpals 8, metacarpals 5, phalanges 14. Here, because we talk about this sesamoid bone, that is patella, right? That is patella, which is also the kneecap, right? So this is extra here. So tarsals, right? Carpals are 8, but tarsals are 7. Carpals are 8, tarsals are 7. So because we have 2 hind limbs, so 7 into 2, 14. Metatarsal will be same, 5 into 2, 10. And phalanges again 2, 3, 3, 3, 3. This is the phalangeal formula. So 2 into 14, it will also be 28. Okay? It will also be 28. Right, bache? So tarsals, they are the ankle. Right? Ankle bones. Ankle bones. Okay? This is the ankle bones. Metatarsals, they are 5 in number. So that's what you have to remember. So ventrally, kneecap is present. So patella is what? It's a sesamoid bone. Patella is what? Patella is what? It's a sesamoid bone. Right, bache? So it's a kind of irregular bone that is totally embedded in the tendon. Okay? So it is the largest sesamoid bone in the human body, patella. It is a sesamoid bone because it is irregular. It is completely embedded in the tendons, right? And tendons, you know that they attach bone and muscle. Muscles are attached to the bone with the tendons. So it is the largest sesamoid bone of human body. So firstly, compare this thing. Understand this maths. Understand this maths first. Yes, understand this maths first. Okay? Done, students? 30 bones and all. Radius and ulna clear. Right? So sesamoid bone is important. It is the knee cover. It is present ventrally and it is the largest sesamoid bone of human body. And moreover, there is one more point regarding this, that it is the patella is the only sesamoid bone of hind limb. Only sesamoid bone of hind limb it is. It is the only sesamoid bone. Sesamoid bones are the irregular bones. They are deeply embedded in the tendons, bache, right? So do write about it. So this is about the names of the form. This is about the name of the bones. Firstly, remember that. Then I'll show you the structure as well. Ye dekho. This is humerus attached to the girdle. We'll come to the girdles later. So baki ye dekho, humerus, radius. This is radius, right? And this is ulna. So radius and ulna, radius and ulna. You have the carpals here, metacarpals here, then it is the phalanges. So ye dekho, right? So there is no need to remember the name of these bones. Okay, so carpals, metacarpals, 5 in number, and these are the phalanges. So ye dekho, here also sesamoid bone is there. You can check it, but yes, you should know about the patella. So ye dekho, 2, 3, 3, 3 or 3. 1, 2, 3, 1, 2, 3, 1, 2, 3, 1, 2, 3, 1, 2. Okay, 1, 2. This is for the hind limb. See this, thigh, femur, knee. This is ventrally, this patella is present. Only sesamoid bone or the largest sesamoid bone of the human body. And only sesamoid bone of hind limb it is. This is the tibia, right? This is the tibia. And this is the fibula. So tibia is thicker, fibula is thinner. right? Remember this, right? So tibia is the thicker one, okay? Okay. So it is the larger bone of the leg. It is the larger bone of the leg, right? It is the larger bone of the leg. Yes. So tibia is thicker, fibula is thinner. Okay. So tibia is also known as the shin bone. It is the one that bears the body weight. Okay. It is the one that this is the shin bone, and it is the one, it bears body weight. It bears body weight. Am I clear? So students, here tibia and fibula, right, towards the lower side, they join with immovable joint. Remember that. So kneecap is this patella, but there is something called knee joint as well. So when you talk about the knee joint, how that knee joint will form, bache, here your tibia, your patella, and sorry, let's start with femur. Femur, patella, and tibia. These three articulate. So remember, fibula will not articulate here. Fibula will not articulate here. So when you talk about the knee joint, it is having tibia, femur, and patella. It is having tibia, femur, and patella. It is having tibia, femur, and patella. There is one trick here, right? With the help of that, you can remember this. Okay, but firstly, write it down. Do it. Yes. Write it, please. Done? Dekho, this is your femur. It is the longest and strongest bone of human body. Longest and strongest bone of human body. Longest and strongest bone of human body. Patella is sesamoid, tibia and fibula. Now in knee joint, matlab this is my trick. This is bad, but theek hai, this is my way to remember it. Femur, tibia. Femur, female. No, two females cannot stay together. So that's why here femur is included. So femur, fibula. Fibula is also female. So femur is like, if I am there, I will not accept fibula. I will not accept fibula. So femur, patella and tibia, they are involved in the knee joint formation, but your fibula is not there. Tibia is there, but fibula is not there. So this information is also important. This information is also important. Sorry, this is my way to remember. Baki, ye dekho here. This is the femur, the longest and the strongest bone. It is attached with the pelvic girdle. Pelvic girdle, this hip girdle, which is quite wide in the females, narrow in the males. Why? Because females, they need to accommodate the baby, they need to give the childbirth. So when you look at this part of this girdle, this part, as I told you already, they have two coxal bones, also known as innominates. So now we will discuss about the girdle. Let's start with the pelvic girdle. Okay, let's start with the pelvic girdle here. So when you talk about the pelvic girdle, hip girdle it is, right? So as I said, coxal bone is there. So each coxal bone is formed by the fusion of, again I'm repeating, it is formed by the fusion of ilium. It is ilium, ischium and pubis. These three will fuse, they will form this bone. So this is one, this is another. So remember, this girdle articulates with vertebral column as well. This girdle articulates with vertebral column as well. Jaise ye dekho, ilium is attached to the sacrum part here. So it articulates with the vertebral column. Pelvic girdle, pectoral girdle will not do it. And here these two coxal bones, they are attached. What is present there? Pubic symphysis. What is present there? Pubic symphysis. Yes. What is present there, bache? Pubic symphysis. Write it down. Write it down. So what is it? It is also the fibrous cartilage. It is also the fibrous cartilage. Yes. It is also the fibrous cartilage. Okay. 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 ठीक. So this is the pubic symphysis. It is also having the fibrous cartilage. That's what you have to remember. Baki, at the ilium is the largest bone here, right? Ilium is the largest bone here, and ischium is basically thickest and strongest. You know, this ischium here is thickest and strongest. So when we sit, our body weight, right? When we sit, our body weight, this ischium is going to bear our body weight, right? This ischium is going to bear our body weight. So bache, at the junction of ilium, ischium and pubis, acetabulum is formed, right? What will form? Acetabulum is formed at the junction of ilium, ischium and pubis. Jaise ye dekho. This is your ilium part, then ischium and pubis. It is named as well. This is ilium, right bache? Ischium. This is the pubic part. This is pubic symphysis, fibrous cartilage it is. So they are formed. Their fusion forms a junction. What do we call it? We use the word acetabulum. What is the word that we use here, bache? At the junction of ilium, ischium and pubis, acetabulum will be there. Right? What will be there? Acetabulum will be there. It is formed at the junction of ilium, ischium and pubis. Right? Ilium, ischium and pubis. Clear, bache? Clear. So basically this femur, it articulates here with the hip girdle at this acetabulum. That's why I'm telling you this name. Jaise ye dekho. Look at this diagram. So this femur will attach here na, to this girdle. So here the acetabulum, where it will articulate. That's what you need to remember. Right? So ilium, ischium, pubis, at the point of fusion of above bones, there is a cavity called acetabulum to which the thigh bone articulates. Important it is. To which the thigh bone articulates. Important it is. Right? So two half of the girdle meet ventrally to form the pubic symphysis containing fibrous cartilage. Right? Containing fibrous cartilage. Understood? Understood? Arey haan, how can I forget? Now bache, we have also completed the rib cage na. We have also completed the ribs na. So ribs ke case mein, one thing, matlab they are the flat bones. Your ribs, your sternum, scapula, they are flat bones. So when you talk about the ribs, they are bicephalic. This is the word, this is the flat bone and we use the word bicephalic here. Cephal word is associated with head. Now in the ribs, you will see, what will you see in the ribs? What will you see in the ribs? You will see kind of two heads here. What will you see? You will see kind of two heads here. Two heads here. And that heads, they articulates with the ribs. Right? So bicephalic ribs we have. What kind of ribs do we have? Bicephalic. You can write down this word. Okay. So ribs have two articulation surface on its dorsal side. That's why they are bicephalic. Okay. Two articulation surface on their dorsal side. That's why it is bicephalic. That is why it is bicephalic. That is why it is bicephalic. Now let's talk about the shoulder girdle. Now let's talk about the shoulder girdle. Right? Shoulder girdle, that is pectoral girdle. That is what? That is pectoral girdle. So you have clavicle, you have scapula. Clavicle is the collarbone. But yes, it is the beauty bone. Just in the like those who exercise a lot and those who are very weak, in their case, this bone is quite prominent. Na? It is also known as the beauty bone. Females, they really work hard a lot. They really work hard a lot in the gym to get these, get them, you know, to see for their visibility basically. So collarbones, clavicle it is, and this side, right, dorsal side is having the triangular shoulder blade. Okay, it is the shoulder blade. It is the shoulder blade. So just like first pair of ribs, clavicle will also articulate with the scapula, and it has to attach with the, it's important that it should attach with the, clavicle will also attach with the sternum. I'm sorry, clavicle will also attach with the sternum just like ribs, and clavicle and scapula, they are also attached to each other. they are also attached to each other. Now this is what we need to understand. So first of all, look at the position. Ye dekho. This is your clavicle. Just like first pair of ribs. Just like first pair of ribs, this clavicle is also attached to the manubrium of sternum. And it is also articulating with the scapula. Look at the scapula part here. It is towards this side, scapula. Look at the clavicle and now look at the scapula. Right, just see, I'm standing here, this side, this is the scapula. So clavicle and scapula, they also articulate with each other. Clavicle and scapula, they also articulate. Look at the scapula, it is also the flat bone, it is the shoulder blade, it is a kind of triangular blade. Right, bache, write about it. It is also important, write about it. So it is triangular, it is on the dorsal surface of thorax. It is on the dorsal surface of thorax, in between 2nd to 7th ribs. Matlab it is about its position, okay? In between, right, it is present in between 2nd and 7th rib. It is present in between 2nd and 7th rib. Matlab its position will start from the 2nd and 7th rib. 2nd and 7th rib. 2nd and 7th rib. Understood? Understood? Sure? Yes? Now, the scapula and clavicle, even they need to articulate. Overall, this girdle will articulate with the forelimb. Overall, this girdle will articulate with the forelimb. But this clavicle, the collarbone and the scapula, even it needs to articulate. So when you look at this dorsal surface, when you look at this elevated ridge, so here this elevated ridge, it also forms a kind of articulation surface for the clavicle as well. So what do we call it? We call it as acromion process. Matlab your scapula will have acromion process. Your scapula will have acromion process where the clavicle articulates. Scapula, that elevated ridge, will form a process, acromion process, where the clavicle articulates. Where the clavicle articulates. Understood? Where the clavicle articulates. Ye dekho. Clavicle and scapula, they are articulating. It is the acromion process. What's that, students? It is the acromion process. What's that? It is the acromion process. Understood? Acromion process. Now, this, you know that this is the forelimb. This is the humerus. It is also articulating with the shoulder girdle. So, it is also articulating with the scapula. So, here what will you see? You will see glenoid cavity. Glenoid cavity. So do not get confused with glenoid cavity and acromion process. So both are there in scapula, and acromion process is the place where the clavicle will articulate, and glenoid cavity is the place where the humerus, the head of the humerus will articulate, right? Where the head of the humerus will articulate, right? So this is also a movable joint. This is also a movable joint, right? This pivot joint is there in between atlas and axis. Cranium bones are having the sutures. So we will discuss about it. Okay, we will discuss about it. So ye dekho. So this is the glenoid cavity in scapula where the head of humerus articulate. This is the elevated ridge having acromion process where the clavicle will articulate. So clavicle and first pair of ribs, right? They are articulating with the manubrium of sternum. They are articulating with the manubrium of sternum. Okay, manubrium of sternum. So this is all about the girdle. So scapula is the large triangular flat bone. Its position is in between 2nd and 7th ribs. Done. Theek. So this is all about the girdles, bache. Now we will talk about the joints. We'll finish it. Then obviously the disorders, and the chapter will be over. So in next 20 minutes, we have to finish it. Okay. In next 20 minutes, we have to finish it. Chalo. So joints, they are very important for all type of movements, right? Including the bony parts of the body, right? So locomotory movements are no exception. Locomotory movements are no exception to this. So joints, they are what? They are the point of contact between bones or between bones and cartilages. They are the point of contact between bones and cartilages. Take. So force generated by muscles, it is used to carry out the movement. How? That is through the joints. So joints act like a fulcrum. Joints act like a fulcrum. So in the paper, they can give you the statement. Muscles act like a fulcrum or joints. It is the joints. Easy it is. Easy it is. It is very, very, very easy. Okay, now the mobility at these joints, it vary. It depends upon different factors. जैसे कि what kind of joint it is. Firstly, fibrous joints. They will not allow the movement at all. In the cranium, we have seen that in the suture, right? So there are, let me write it. We have fibrous joint. We have cartilaginous joint. We have synovial joint. We have synovial joint. So when it is the fibrous joint, when it is what? When it is the fibrous joint. So what's that? It is immovable kind of joint. It will not move at all, right? It is synarthrosis. It is synarthrosis. It is immovable type of joint. What kind of joint it is? Immovable type of joint. Cartilaginous, a little movement is allowed here, synchondrosis. It is known as synchondrosis. Synovial, it is freely movable type of joint, right? It is diarthrosis. What's that? It is diarthrosis. These are the names here. These are the names here, right? That pivot joint was the example of synovial joint. So fibrous joint comes. So again, here examples are important. What is important, bache? Examples are important. So in cartilaginous joint, that is synchondrosis, the bones involved are joined together with the help of cartilages. So the joint between adjacent vertebrae, just I gave you the example of intervertebral discs. I gave you the example of intervertebral disc, fibrocartilaginous joint it is. Then, yes, because it will allow the limited movement, right? It will allow the limited movement. So this is the example of cartilaginous joint. Is that clear? This is the example of cartilaginous joint. Is that clear? Is that clear? So they are slightly movable. They are slightly movable. And that pubic symphysis, that we have discussed in pelvic girdle, that is also the example of cartilaginous joint. Okay? That pubic symphysis is also the example of cartilaginous joint. Please write down. Pubic symphysis is also the example of cartilaginous joint. The joint which is in between the sternum and ribs is also the example of cartilaginous joint, right? The joint which is present in between sternum and ribs, that is also the example of cartilaginous joint. Understood? That is also the example of cartilaginous joint. Now, bache, synovial joints, movable joint, right, bache? It is diarthrosis. So it is having there is the presence of fluid-filled synovial cavity between the articulating surface. जैसे ये देखो. Look at this. This is the bone part here. And look at the patella, kneecap here. The only sesamoid bone of your hind limb. This is the another bone, right? So in between, this is the articulating surface. You will see the articulate cartilage here, right? What will you see? You will see a kind of space here, kind of cavity here. Read it. So there is a fluid-filled synovial cavity between the articulating surface of the two bones. This arrangement will allow considerable movement. So this joint help in the movement and many other movements, bache. This is the synovial joint. So here in this synovial cavity, जैसे there is the articulate cartilage. देखो, here you have the articulate cartilage ना. Here you have the articulate cartilage na. So in between the articulate cartilage, this space is actually the synovial cavity lined with synovial membrane having the synovial fluid, right? So the inflammation of this synovial membrane can result in, it will be there in rheumatoid arthritis. Rheumatoid arthritis, you know na, it is the autoimmune disorder. Autoimmune disorder. So there is the inflammation of synovial membrane there. Inflammation of synovial membrane there. So look at the synovial membrane part here. Okay, so we have the joints here. We have the joints here. जैसे ये देखो, ball, ये देखो, this part, ball and socket joint, right? This is also your ball and socket joint. Ball and socket joint. This is hinge joint. Hinge, जैसे laptop hinge. Laptop, we fold it like this na. Hinge joint it is, right? So here also hinge joint, right? Fixed joint. बाकी ये देखो, ball and socket joint. The pivot joint is, you know, na, swivel joint, pivot joint. That is also the example there, right? Or saddle joint. Chalo, let's discuss the examples. So firstly, ball and socket type of joint. Ball and socket type of joint, right? So ball and socket में head of humerus and glenoid cavity. of humerus and glenoid cavity, and head of femur and acetabulum. Head of femur and acetabulum. This is the example, right? This is the example. So shoulder joint and hip joint is the example, right bache? Next is the hinge joint. Next is the hinge joint. They all are the example of movable joints. So hinge joint, it is in between. Hinge joint, it is in between. This is the elbow joint and the knee joint. है ना? It is the elbow joint or the knee joint, and even बच्चे the joint, देखो joint between phalanges. It is the joint between phalanges. Are you getting my point? So elbow joint, knee joint, and the joint between phalanges. Elbow joint, knee joint, and the joint between phalanges. So elbow joint, knee joint, and the joint between phalanges. So pivot joint is in between. Yes, pivot joint. Between atlas and axis, or between radius and ulna. That is the example of pivot joint. That is the example of pivot joint, right? That is the example of pivot joint. And there is a saddle joint which is there in the carpals and metacarpals of thumb. So my thumb is sad. That's how I remember. That's how I remember. Saddle joint. It is between carpals and metacarpals of thumb. It is between carpals and metacarpals of thumb. What is it? It is the of thumb. It is the saddle joint. ये देखो, it is marked here, saddle joint. Right, saddle joint, ball and socket clear. So one is बच्चे gliding joint also. Which is also known as plane joint. So here you guys can see, this is what they are highlighting when they are telling the gliding joint, that here you can say that gliding type of movement will be there. So it is in between carpal bones, in between tarsal bones, okay? Because these are the tarsals now. So it is between carpals, the type of joint is gliding. Between tarsals, the type of joint is gliding. So that's it. That's all. Do remember the examples. It is important. Take. It is important. Okay. Now let's discuss about the disorders. So I hope up to this part everything is clear. Right? So study of joint is called arthrology. Arthrology. Study of joint is what? It is arthrology. Study of joint is what? It is arthrology. What's that? It is arthrology. Right? So disorders में बच्चे देखो, myasthenia gravis, it is autoimmune disorder. It affects the neuromuscular junction. I will not add much things here. At least remember the disorder and with which system it is associated. This is what you have to remember. So it is the disorder which is affecting the neuromuscular junction. So the fatigue is there, weakening and paralysis of skeletal muscle. Muscular dystrophy. Bache, in the case of muscular dystrophy, there is the issue with the dystrophin gene, the largest human gene. So progressive degradation of skeletal muscles due to genetic disorder. Tetany. And let me tell you again, tetany and tetanus, these two things are different. Tetanus, tetany, these two things are different. So tetany is the rapid spasm, means wild contraction in muscles. And how is it possible? Due to the low calcium level in the body fluid, right? So of course it is also related with the hormones, because low calcium is there in the fluid, matlab your parathormone, which maintains, Collep's hormone maintains the calcium level. Now, when it is low, it increases that, right? So means it is not that available. So tetany is the rapid spasms in muscles, and tetanus is what? Yes, tetanus is, right? It's a bacterial infection that can also result in spasms, but it is caused by the bacteria Clostridium tetani. Okay, Clostridium tetani. Okay, baki fir arthritis, there are different types of arthritis. Arthritis in simple word is inflammation of joints only. But yes, there are different types of arthritis. Arthritis in simple words, it is the inflammation of joints. But there are the type of arthritis. There are different types of arthritis. जैसे we have gouty arthritis, rheumatoid arthritis और osteoarthritis. So rheumatoid arthritis is the second most common type of arthritis. First one is osteoarthritis. Right, so rheumatoid arthritis, as I told you, it is the inflammation of synovial membrane. It is also an autoimmune disorder. So gouty arthritis mein bache, what is there? There is excess formation of uric acid or inability to excrete it. Right, so there will be excess of uric acid. So gout, it is also known as gathia. So excess of uric acid, or the body is not available to eliminate it. Okay. Okay, so these uric acid will get deposited in joints. Okay. Okay. Rheumatoid arthritis, I told you, it is due to the presence of rheumatoid factor. Right. It is also autoimmune. That's important. It is autoimmune disorder, and it is due to the presence of RA factor. RA factor is bache rheumatoid arthritis factor. It's a type of IgM immunoglobulin. It's a type of IgM immunoglobulin. In my family, 2 members are affected with it. 2 members are affected with it. So IgM test will be done. Matlab this will be checked in the body, right? Right. So baki osteoarthritis, it is osteo mane bones. So it is age related, right? Females, they are most likely to affect with it, right? So after menopause, because the level of estrogen will decrease now. So that is why bone mass will also decrease and the fracture will increase. So this is all about the disorders here. This is all about the chapter. Yes, we have completed it, and yes, I have shared all the required information. So now you just need to practice the questions from this particular part, and I will share the PDF in the Telegram group as well. Okay, I will share the PDF in the Telegram group as well. So do not forget to join our Dream Crash Course batch, bache, where the proper revision, proper test, proper practice will be there. You can use this coupon code Ambika10 in the description box of this video. You will get the link for the batch. Okay, so the next chapter that we are going to discuss is body fluid and circulation. Okay, we will discuss the body fluid and circulation next, and day after tomorrow, I'll keep a practice session also. Okay, I will keep a practice session also. So stay tuned, revise it. Day after tomorrow, I will keep the practice session. We will revise the PYQ. So I will share the PDF in the Telegram group. So students, if you like the lecture, if it is beneficial for you, please do not forget to mention it in the comment section. Okay. Okay. So do not forget to mention it in the comment section. So thank you so much. Take care. Revise well, study well. Still you have time to complete your syllabus and to revise it. But yes, you have to follow a smart way. So you can watch our videos on the on our channel, that is Unacademy NEET English. Me, Yawa sir, Wazeem sir, we keep posting that kind of videos. So you will get a proper strategy as well. So take care. Bye bye. Thank you so much.